Дорогие друзья, дорогие коллеги, 10:15 мы можем начинать. Я приветствую всех собравшихся в этом зале. Кто не знает, меня зовут Владимир Попов, я научный руководитель федерального центра. And from now on I'll proceed in English because I was asked to do it in English. Our conference has an international dimension, so English is an international language of science. So dear colleagues, dear friends, I would like to greet everybody at the opening of our School for Young Science in the field of structural biology. It's a satellite event accompanying our traditional conference for young scientists in microbiology, which we organize from time to time. It is the 13th event now on, and I hope that probably this school in structural biology will be a good tradition now just starting. So 2 years of COVID pandemic and current geopolitical situation, it doesn't benefit the international exchanges, person-to-person contacts, and I am very happy that we managed to assemble such a nice program with such distinguished speakers from all over the world. I'm very grateful for them. Some of them are located in quite distant time zones, so it's real sacrifice for them to deliver their lectures live. It will be, in United States, it will be early dawn or even middle of the night. So my compliments go to these courageous people. And seeing is believing, as you know. It goes without saying that structural biology is one of the main pillars of the modern life sciences. Without our science, it will be impossible to understand the intricate mechanisms of the nanomachines working in our cells. It will be impossible to design new efficient biocatalysts. And of course, structure-assisted drug design is a well-known area, well-developed. We just experienced a cryo-EM revolution. It's a very dynamic field, and right now we're monitoring how the artificial intelligence or deep learning machine algorithms are penetrating structural biology. And I want to congratulate the organizers of this conference who managed to compile a very impressive program that spans all areas of structural biology, from conventional MX to free electron lasers to NMR, cryo-electron microscopy, modeling, artificial intelligence. So I look forward to the very interesting and illuminating lectures, and I am sure that they will be to the benefit of the young generation of structural biologists. Not so numerous as I see in the audience, but I hope that very many people are at their desks, at their notebooks, monitoring our conference. So I don't want to take too much precious time from the lecturers, so probably that's enough for the beginning. Once again, I would like to say that we are looking forward for the very interesting day of interesting exchanges, discussions, and very important illuminating lectures. So I would like to pass mic to the first moderator of our morning session, to Eugene Maximov, yes, who will introduce our first speaker. I sincerely hope that our internet lines will stay open and will fit and won't spoil, won't ruin our conference. Okay, thank you very much.
Thank you, and I would like to say thank you to our hosts from the organizing committee, and we shall not waste any time, and because we have very dense program and a lot of very interesting lectures. And our first speaker is a famous scientist in the field of structural biology who made really a lot of developments and discoveries in the field of research of photoactive proteins and ion transport proteins. And during his lecture, you will know about new fields of structural biology, which is now rapidly developing and related to time-resolved experiments. So please welcome Valentin Ivanovich Gordeliy. I hope he's here, and now we can begin.
Demonstrate my presentation. Just a moment, please. So thank you very much for the invitation. It is not a great honor, but it is also a great pleasure for me to give a talk here. Moreover, there is something to tell which might be of interest of young scientists and maybe not only young scientists. So I will tell you how to see the mechanisms of protein, of a protein, molecular mechanisms, how to elucidate them, and certainly to know exactly what is the mechanism of protein function. We have to know the structure of this protein, high-resolution structure, not only in the ground state, but also in active states. And normally there are several key intermediate active states of the protein, and we have to be able to determine the structures of the intermediate states in the best all intermediate states before we can provide reliable conclusions on the mechanisms. Today I will tell you two things. I will mention some methodologies which allows one to see how protein is working in course of its function. One approach is to trap the key intermediate states, normally with so-called cryo-trapping, and the other approach is to do time-resolved crystallography. Today I will not bother you with the details of technical details. I will give you the references. Fortunately, there are proper references, and if one would like to go deeper to methods, one can do this easily. This I will mention, the references I will mention in the end of my talk. One more thing that, of course, would be nice to do an overview of how people are studying
do you change your slides?
the mechanisms of the proteins using X-ray crystallography, and crystallography in general, but there are not so many examples. What I will demonstrate today is really unique. To my best knowledge, there are no such data on any other protein, especially when we speak about membrane proteins, and especially when we speak about proteins and mechanism and related mechanisms, where we have to have highest quality of the data.
Valentin Ivanovich.
Yes.
Your slides did not change.
Yes, I will. This is just an introduction.
Sorry.
If we speak about true mechanisms, a deep understanding of the mechanisms, that in some cases it's possible only when resolution is true atomic. True atomic resolution is resolution around 1 angstrom, which is normally difficult to obtain. There are just a few examples of such resolution, and certainly there are even less examples where you have structural resolution about 1 angstrom for the ground state, not only for the ground state, but also for the intermediate states. So today I will describe illustrating time-resolved crystallography and cryo-trapping approaches. I will speak about this level of resolution. So let's go to the next slide. So I will focus my talk to rhodopsins. Rhodopsins before this century were very important but very exotic, very rare proteins which were found only in archaea, and there were only 3 examples of different types, functionally different proteins. But already in the beginning of 1971, there was a big discovery. At the time, it was not possible to predict how big is this discovery. People discovered bacteriorhodopsin, an archaeal proton pump. But then in this century, in the beginning, it became clear, especially because of metagenomic approaches, that there are so many rhodopsins in all domains of life that I present here a planet, and this is from the cover of a book. And why we have green, it's of course plants, but in ocean, this color which corresponds to bacteriorhodopsin, of course, this color should be also somehow modified. But what I wanted to show in this business, that in ocean, the major light harvesting proteins are rhodopsins. So you can imagine how fantastic is their role in our life at our planet. But in addition, rhodopsins triggered one of the highest and most technologies and long sought technologies, optogenetics, But today I will speak about this old story and then a little bit about new story of bacteriorhodopsin. During many years, bacteriorhodopsin, here I show this membrane protein schematically, 7-alpha helical protein, which pumps proton. This protein pumps protons against a huge gradient, electrochemical gradient, up to 0.2 volt at membrane. And if we take into account 5 nanometer thickness in average of membranes, then you can imagine how strong is the field. So this protein catches by chromophore retinal energy of light, absorbs photon, and uses this energy to pass proton against the strong gradient. And during many years, this was a big puzzle how this protein does. And certainly, this is true that this, at least at certain time, was a racing for Nobel Prize in which Richard Henderson, who finally received Nobel Prize in 2017, recently for cryo-EM, but he developed cryo-EM working on bacteriorhodopsin. This probably not everybody knows. So bacteriorhodopsin in general, one can say, is a king of membrane proteins, and this may be like a mystery. Mechanism of proton pumping was or proposed mechanism also based on high resolution structures. Also intermediate states were very controversial. 3 different major groups working in the field came with different structures of the intermediate states and with different conclusions about the mechanism. So let's go faster here. I think that you see what I show. Here is the ground state. When proton is absorbed, then the protein undergoes several functional intermediate changes and then returns back. And then photocycle can be restarted when a new photon comes. The total length of the photocycle in this nice bacterial rhodopsin is about, let's say, 20-30 milliseconds, depending on conditions. And there are key intermediate states in the beginning, which are important to understand the mechanism. Is the K state and L state and M state. It was always considered that a key to the mechanism is the understanding what is going at this early, so-called early intermediate states. So to get a mechanism, one has to know the structures not only in the ground state, but also in the intermediate states, as I was telling you. To do this, one has to use X-ray crystallography, one has to have fantastic crystals, and this was our case. We show here the crystals which we were able to grow, and this was a big development of the crystallization to obtain these crystals. So I will describe qualitatively a major idea of cryo-trapping of the intermediate states in the proteins. So we have we have a photon, and we can excite, trigger the cycle of the changes. And what is important, these intermediate states, and there's a big log, they correspond to to the key functional states. And you see, for example, then between L and M states, the proton is going out of the protein to the extracellular part of the membrane. So, but when we trigger the photocycle, certainly protein will go through the whole photocycle, how to trap the intermediate state. And the idea is very simple. So this, as you see, fast intermediate states. To avoid passing the whole photocycle, we freeze the crystal, and then only under certain temperature we excite crystals with a laser beam. I'm simplifying, not giving any details here, but the idea is very simple. We have to use temperature to block protein at a certain step. So we trigger, excite protein, and the protein goes along the photocycle, but due to the low temperature, it cannot overcome energetic barrier to go to the next step. So we normally trap the intermediate K, at 110 Kelvin, and then move again to 100 Kelvin to avoid radiation damages, to trap for longer time this intermediate state. And to trap L, we increase temperature in the other experiments, and then temperature to 150, 160, and then photocycle is blocked, the L state, and so on. And the M state is trapped under room temperature. Again, I will not go to the details, we will not have time. So absorption, but how to control whether we trap a proper state? We can do microspectroscopy with single crystals. Later I will show setup, and we see the absorption spectra, and this absorption spectra maximum has a certain wavelength, and the intermediate states, as you see, normally have different, sometimes very different absorption wavelengths. And here they are shown. Here, for example, M state is just 410, and here is 590. So you can easily control whether you have a proper procedure to trap the intermediate state. The story is not so simple. It's not possible in any experiments which has been done before. There was no an example where one has, except our experiment with the other proteins, where one could have 100% of the occupancy of the intermediate state. Normally you have the ground state and the intermediate state, the mixture. But you can, doing a proper treatment and knowing the ground state with really high resolution, you can, of course, subtract from your data the structure of the intermediate state. So let's go further here. I describe very recent results, and we were working on this project during 20 years, more than 20 years. And I was working with different students and postdocs, undergraduate students, and many of them, they did nice contributions like Vala, and before they were, the story was started before, there were some other students like Ruslan Efremov, etc. Therefore, I'm very grateful to them, but I would like to say that all what I will demonstrate, all the intermediate structures had a resolution about 1 angstrom. And this is something which is absolutely unique. And why it was necessary to have, in this case, to elucidate the mechanism of the transport. Certainly, when we speak about proton transport, we have to take into account that proton transfer should go through hydrogen bonds. Hydrogen bonds, many of them are mediated by water molecules. Protein is a dynamic object, and water molecules moves, and some amino acids moves also significantly. Therefore, with lower resolution, you will not see, as it was in prehistory, people were not able to see the whole picture. They saw only the fragments of what one needs to see. And these fragments, unfortunately, did not allow to reconstruct reliably the mechanism and even pathways for proton translocation. I will not have no time to go in details. You can read in our papers about methodologies and about this story. This story was published in this year, and before there were several key in sense of the importance. Our paper on radiation damage, on twinning of the crystals, and these problems has to be solved in this case to obtain these results. So now what do you see? You see these quite bold lines. Let's show the L state of the protein. You see this is a representation of the whole structure of the protein, a part of the structure for the ground, K and L and M states. Let's take into account, let's consider the L state. You see that this hydrogen bond, linear chains, they pass through the whole protein. This is absolutely unusual and unexpected. Moreover, we show here that this cytoplasmic part of the protein, which was always a puzzle, how proton can go through this part. Here we have also water molecules and hydrogen bonds in the L state. How it's possible? Again, leaving the details, I would like to say that the properties of linear chains, or as we know from physics, dimensions of the object can considerably change the properties of this object. And one, here you see only water molecules. They create a linear chain of hydrogen bonds which penetrates this hydrophobic region of the cytoplasmic part of the protein. And let's say this is not very exact and very correct, but one can also use this to describe qualitatively what happens. So we see that with linear hydrogen bonds, we can have water molecules in oil. As we know from school, oil and water cannot be mixed, but it can be mixed when we have this nice picture. when we have linear, one-dimensional, so-called proton wires. And our main conclusions were that proton wires, this is the core of the mechanism of proton transfer. But proton wires in the bacterial rhodopsin, they are not only pathways for proton transfer, but they are also provide signaling over long distances in the protein. And this is something which of high interest. And this is a new thing in biology, that one can pass signals over long distances using these one-dimensional hydrogen bonds. Again, why we came to these conclusions, it takes some time to explain, but let me try to do this. I will pass this one of the slides. Let's go. Let's imagine that there is a one-dimensional here wire, or proton wire, or hydrogen long hydrogen bond chain. And then let's imagine that we changed the state of an amino acid, for example, pK or charge, in one of the ends of this chain. That what is happening, I can transfer the information about this event over long distances to, when we speak about bacterial rhodopsin, to the so-called proton release spoken. It was a big puzzle how proton is released. What is the mechanism of proton release to the extracellular part? So when we see this movie, you will understand. So it's not necessary for a proton to go through the whole sequence of these balls, the so-called Maxwell pendulum mechanism. Just you change something at one of the end of the wire, and the information will be passed through the wire to the target. And target in that case was a proton release, and proton will be released. So I will not really go to any details, but I would like to say that what we revealed with cryo-trapping, the mechanisms, but the mechanisms appear to be fundamental. They are based on deep quantum mechanical properties of linear chains of hydrogen bonds and so-called low barrier hydrogen bonds. But let's go further. Let's speak about time-resolved serial crystallography, which we use. And this is maybe major message can be given in a few words, and then we can stop the presentation. You can read, you will be able to read the details of this work soon, because it's nearly accepted in one of the journals which everybody know, etc. This can be read in this journal, and you will see all details. But what are the results? what can one achieve doing time-resolved serial crystallography at, probably you would immediately continue at X-files. No, not X-files, synchrotron sources. So we achieved structural resolution about 2 angstrom with our crystals, but I will speak now about another rhodopsin inward pump, which transfer protons along electrochemical gradient. So, and what is time resolution? Time resolution is sub-millisecond resolution. Due to the fact that that machine, which was DAISY synchrotron and EMBL beamline P14, we did this experiment there. And this is not, still the synchrotron source is not fourth generation, but the beamline is perfect due to so nice crystallographer like Gleb Bourenkov and his colleagues. So we were able to achieve sub-millisecond, 0.5 millisecond resolution, but the limitation was not our crystals. They unfortunately do not have yet so-called Frau detector, which is fast. So we used quite slow detector, and this was a limit of the detector. So in the nearest future, we expect that we will be able to achieve 100 microseconds. And if we say that we are not responsible, what I'm saying now to you, but our dream is 10 microseconds. And what is 10 microseconds? It means that we can cover with synchrotron source, of course, if you have good or excellent quality protein crystals, we can really cover nearly all biologically relevant processes in the proteins. So let's go to the next slide. Here I just demonstrate our very old paper 2006. Ruslan Efremov at that time was working with me. And these experiments, first of all, he prepared a setup to be able to do spectroscopy with the crystals and also time-resolved microspectroscopy with a single crystal. You see this crystal and you have this scheme. And here there is a light, here is our crystal, this is, and then the camera are detecting spectra. You can have flash photolysis set up or just measure spectra on the crystals, but also one can do, and Ruslan was able to set FTR time result with the crystals. So this was nice, and this is very important. When we speak about time resolved, we have to do time resolved. First, we have to do time resolved spectroscopy with the crystals. Then we will know when, where, at what time what time is significant for our time resolved X-ray data collection. And I have to mention that photocycles, kinetics of photocycles can be different in some parts, very different in the crystals and in solution with the protein in solution. Let's go further. Just a few minutes, if I still have. This paper will be published soon. And why this work was for us extremely important also. It was important because, okay, we have a fantastic mechanism which is based on quantum mechanics and deep properties of linear hydrogen bonds. Very interesting, but how general are these mechanisms, whether they are applicable to the other proton transport proteins and whether they are universal also for some other proteins, not only this, and therefore we extended our work to two other types of the proteins, and one is also rhodopsin, one of them, but inward pump. Of course, the structure is very different and the function is very different, and therefore we were interested whether still we see this linear chains of hydrogen bonds, proton wires, etc. And in the right up corner, there is a photocycle. And you see that there are K, L, and M intermediate states and the ground state. So we were able to obtain highest quality data and also time-resolved data with this protein for all the intermediate states. I really do not know any other example in crystallography where all key intermediate states were studied and the structures were solved to true atomic resolution or nearly true atomic resolution. So let's go to the next slide. Here I show just
Валентин Иванович, у вас 2
electron
минуты.
densities.
Валентин Иванович, две минутки.
I'm coming to the end, do not worry. And this is really last slide where I demonstrate we see again, look, linear chains of hydrogen bonds which are penetrating the protein, and how it works, how they serve for signal and proton transport. This is, of course, a matter of a separate seminar. I will not stop on this. So this work allows us to not only to show the mechanism of inward proton pumping, but also be able to generalize what we found with bacterial rhodopsin. So let's, there was demonstration of quantum mechanics, how one can, proton wires can work. Let's go, I think that the participants can get at least some of the presentations, no problem with ours. So here there are our publications where you can read about cryo-trapping, and in this last work you can, which will be published soon, you can read about time result. And also I would like to say those who are interested what is going at our planet and to get a deeper insight to the methods, also XFEL methods, time resolved methods, you just go to this year published book where you can find not only descriptions of the fields, but also methods and also protocols, which sometimes is a key for young people to enter the field and to be successful from the beginning there. Thanks a lot for your attention, and hope that at least something was useful.
Valentin Ivanovich, thank you very much for your lecture. It was very interesting, and I actually would like to ask you a lot of questions, but we are short on time, so I think we have time for only 2 questions. And if somebody in audience want to ask something, we have a microphone here, but I would like to go first and ask you, you mentioned that structural biology in this field is driving optogenetics, and what do you think is the perspectives of this approach, I mean, in medicine, and what things could appear in next, for example, 10 years?
Good. Just one maybe remark. I was not telling that only structural biology drives optogenetics, but structural biology is a key for understanding the function and also for engineering of optogenetic tools, rhodopsins. This is absolutely true. It means that this is a key approach within for optogenetics, for the development. You can read about what you asked in one of the chapters. There are 3 general chapters in this book, and one chapter is on optogenetics, what are rhodopsins and optogenetics, and also relation to medicine. Just to tell you, at least two hour discovered by us and characterized and structurally characterized rhodopsins now are in preclinical studies. One study is for recovering of loss of hearing in patients, and the other is recovering of vision in completely blind patients. And I would like to say that this is just a small piece of what one can expect, applications of rhodopsin optogenetics to medicine use. Let's
Thank you.
answer in this general way.
Thank you, Valentin Ivanovich. Thank you for your lecture, and thank you for attending in this conference. Now,
Thank you very much.
Thank you. Now we need to go to our next lecture, which will be from Professor Albert Guskov from Faculty of Science and Engineering, Groningen Biomolecular Sciences and Biotechnology Institute from Netherlands. His lecture will be dedicated to method which is now applied to very huge amount of biological samples. It's
hello, dear colleagues. Can you hear me?
Microscopy. Yes, we can hear you. And please start at any time when you're ready. Professor Guskov, are you here?
Dear colleagues,
Yeah,
can you hear me?
Perfect, yes.
Perfect. Okay, nice. Let's start. So my name is Albert Guskov indeed. So I'm a professor of structural biology at the University of Groningen, but I'm also a visiting professor at Moscow Institute of Physics and Technology. So today I will give you a rather brief overview of cryo-EM technique of single particle analysis. And actually I will give you 3 examples how you can use this marvelous technique in, well, addition to, let's say, fantastic time result studies, you can do what Valentin Gordeliy has just told you about. So why are we using cryo-EM more and more? In principle, it's pretty old technique. So it's almost like 100 years old. And already in 1933, a German scientist, Rizhikov, actually came up with already working electron microscope. Already before that, people realized that we can use electrons to study the matter. It's actually quite astonishing that just after the war, these microscopes already were produced on a large scale. So let's say in 1949, Philips was producing several hundreds of these microscopes. But at that time, mostly, of course, people were doing the material analysis. So the soft matter, proteins, still have to wait till 70s or so. So how microscopes look these days is just a box, depending on how rich you are. The box is bigger or smaller, but in principle, inside it's more or less the same thing as here. So you have a column, you have certain electromagnetic lenses, if you like, you have a good detector, etc., etc. So why electrons are good for studies for structural biology and in principle for structural analysis? It's relatively easy to produce. And this is true, let's say, for basically for any source. There are different sources, cold sources, hot sources, etc., etc. But let's say the main benefit probably that it's very easy to focus. So compared to X-ray radiation, for example, which barely possible to focus electrons, well, you just put lenses. You can adjust the current and you can easily focus them on a certain plane. And clearly the benefit is that you have a small wavelength. It's actually way smaller than the wavelength of X-ray radiation. So in principle, you can study even sub-angstrom resolution easily, but well, easily is not going to work because there are too many difficulties and aberrations and problems, etc. But let's say for soft matter, anything close to atomic resolution, which is going to be 1.5 angstrom or better, is actually perfect because we can see all the details of interactions between molecules, proteins, nucleic acids, small molecules, ligands, etc., etc. However, there are also problems with electrons, and one of the problems is very low penetration, especially compared to X-ray, for example. So this is the reason that for transmission electron microscopy, you need to have extremely thin sample. And this is a few hundred nanometers. That's why, for example, if you want to do a study on the cell with tomography approach, you really have to cut it to feed mill it. Otherwise, you will simply not see anything because electrons will not go through. The other very serious problem, and actually one of the main limitations why we cannot achieve even higher resolution, is radiation damage, which is unavoidable. So whenever you have a flow of electrons and it interacts with the matter, so there would be a production of so-called inelastically scattered electrons. And those electrons are really bad guys because they actually produce different kinds of radiation damage, which eventually destroy your sample. And we should not forget actually that the specimen is frozen, and many people claim that cryo-EM is close to native condition, but well, apparently it is not because you're already in the cryogenic mode. So where are we now? What is the state of the art? So you might have heard that recently Thermo Fisher, one of the key players in the on the market, released a new generation of energy filter selectrics, two different types of it, but both are equally good. So what it allows you, it actually allows you to get rid of most of inelastic scattering, and you actually only make very thin, basically it's called zero loss peak. So you really take only the best electrons, so those which actually produce the contrast. That means that even small molecules like hemoglobin, which are basically like 64 kilodaltons, so you can study with cryo-electron microscopy, because historically everyone was like, oh yeah, we can use it for a ribosome, but not for something very small. So now actually you can. And the great benefit also of this stable and zero loss energy filtering, that actually you need way less particles to achieve the same resolution, like in this example. So only with 370,000 you achieve 2 angstrom resolution, whereas with wider slit you need to collect basically 1 million of particles. There are also fantastic detectors now available, like Falcon 4i, Gatan K3 or Polar, and all of them basically allows you to go to angstrom resolution. So you can see hydrogen, for example. There are also now on the market new sample preparation platforms. So you may have heard of Chameleon and Vitrojet, where actually many things are in automatic mode now, and you can really very fast on milliseconds scale freeze your samples. But what to do if, well, this is beyond your reach, because yeah, realistically, only few labs in the world have now access to all these fantastic developments. So what you can do if you're, well, less lucky in this life. So disclaimer here, you still need a decent direct electron detector. Otherwise, it's going to be very problematic to do any good structural biology studies. So, and I'll give you some examples now where we did quite some work using our in-house microscope, which is Thales Arctica 200 keV with K2 camera, which again is not a bad camera. So the first example is solid carrier family 1 of transporters. So very briefly, I will introduce this family. So there are many transporters in humans, and you can separate them into, you can put them in different classes based on what kind of molecule they transport. And we're interested in neurotransmitters, and there are two families, and SLC6, let's say the one which transports dopamine and other very famous neurotransmitters. And there is SLC1, which transports basically glutamate, which is actually a very important neurotransmitter. So there are 7 proteins in this family. We will not go into details here, but basically I want to tell you that misfunction of these proteins have very bad consequences. And the role of these transporters is actually when you have signal transmission from one neuron to another neuron, what happens here, you have a release of glutamate, which is a neurotransmitter, which stimulates receptors. So as soon as it has stimulated the receptor, this excess of glutamate must be removed. Otherwise, you will continuously stimulate your receptors, and that's not the best idea. So basically, these transporters, they ensure that you can pump out neurotransmitter very quickly, and then it's going to be the resting state. Unfortunately, well, sometimes these transporters start to work in reverse, let's say in the case of ischemia, because you have a change of potential on the cell, and actually they start to pump in the neurotransmitter in the synaptic cleft. That leads to the neuronal death. So it's very important to know the mechanism of this work. So our group and many other groups in the world actually spent quite some time studying these transporters and the mechanism using crystallography, EPR, FRET, IFM approach. And we noticed one interesting thing. Whenever we do something in terms of structure, we usually have these transport domains. So you basically have a trimeric structure, scaffold domain, and 3 transport domains which move independently. So whenever we studied those transporters, we saw that they either all in outward facing or inward facing state. And we really wanted to see them in different conformations. So what we did, we went to our in-house microscope, and I will not go into the details of data processing, but we did a smart thing, in my opinion. We did sort of titration. So we studied it in undersaturated conditions. So let's say we have a structure when we have no substrate, we have a structure when we have saturated condition, and then transport domains all in one position. But we also have unsaturated state. And what we actually observed, that some of transport domains are inward and some are outward. And let's say this is done in one sample. So this is another beauty of cryo-electron microscopy, that quite often you can study different conformations already in one sample, which is very rarely observed in X-ray crystallography, for example. And well, as addition, we also did the study with inhibitor. So we could actually resolve quite a lot of states. Some of them were very new, never observed before. Let's say like inward open and some occluded states. Again, I will not go here into the details, but my message here is that actually just with a couple of samples, you can describe an ensemble of different conformations of this transporter. And let's say we actually realized why this transporter needs both substrate and sodium, because it's sodium dependent. Because if you have only sodium, there is a so-called gate of the transporter in the transport domain which cannot close. So it actually behaves the same way as when you use the inhibitor. So inhibitor is bigger than the substrate, and actually prevents the closure of the gate. And when you have only sodium but no substrate at all, you basically observe the same situation. So you really prevents the sodium spill. Another example I'm going to give you is about the transport of cobalamin in Mycobacterium tuberculosis. So I don't have to spend any time telling you how bad this pathogen is. And many people in the world are actually quite sick from it. But an interesting fact that basically all cobalamin on Earth is produced by a limited number of bacterial species. So cobalamin is one of the largest small molecules which are used in biology, and actually synthesis of this is, well, extremely energy demanding and lengthy. So in many cases, actually, organisms prefer to get cobalamin from the environment. So it's easier to scavenge cobalamin than to produce it. And we looked at the genome of Mycobacterium tuberculosis, and what we could see that there are quite a lot of ABC transporters. And we know that, well, there are certain B12 transporters known from the bacteria, but they are not found in the genome of tuberculosis. So there are different candidates proposed, and in principle, the conclusion was that there are some organisms which contain BTU-CDF, and the structure was solved like 10 years ago or so. And there's also some unknown uptake system which was fully uncharacterized. And there actually were several proteins later revealed. So, for example, ECF-CBRT, which first was predicted with bioinformatics approach, and later we solved actually the structure of this transporter. Same for BTM, again, was proposed by Rodionov and Galfand pretty long time ago. Later we solved the structure, and was also the interesting gene RV1819C that was proposed to be involved in the transport of cobalamin. So I will not spend much time on this. So we basically designed a strain which with knockout of all known cobalamin transporters. And what we could then later do, we were adding, let's say, we were rescuing the phenotype by adding different transporters. So the known BTCDF, BTM, but also the proposed RV1819C and the mutant. And we can actually see that all of them work nicely, clearly except the mutant. So they could really rescue the phenotype, and the cells were growing. So let's see. Yeah, so we solved the structure. Here I must actually admit that some of these structures were also solved using Cryos NK3 detector and not to Talos NK2, but some of the work was also done with Talos NK2. So you can see we resolved it, well, still at 3.5, not too extreme, unfortunately, or better. Nevertheless, it was enough to actually analyze the structure. And one of the interesting part of this was that it actually has an exporter fault. So ABC transporters in general can be subdivided into different types, like importers, 1, 2, ECF transporters, but also exporters. And surprisingly, the structure we solved resembles more the transporter than importer. But we showed that actually functionally it is an importer. So it's one of the perhaps first examples where actually you can see that exporter fault can also be used for import of molecules. Yeah, that's just another confirmation. When you use the knockout of E. coli, we can actually rescue a phenotype with this transporter. And when we spend some time analyzing this transporter, we actually realized it has enormous, extremely large cavity inside of it, which is basically around 7,700 of cubic angstroms. So for comparison, if we look at the structure of BTCDF, which was resolved much earlier, this is actually pretty small, so it's around 1,100. So you can imagine that you can fit up to 7 cobalamin molecules, which, well, perhaps it's not even necessary, but what we realized next, that perhaps this is a very promiscuous transporter, and it can transport not only cobalamin, but also other compounds. And it seems to be so. So we did some functional studies where we could show that it can also transport different peptides. And we do not even exclude it can be involved in transporter of some charged antibiotics inside the cell. And yeah, this we can skip. Yeah, that basically it can be a non-specific transporter for hydrophilic molecules, and it seems to be so. The last example I want to give you is on a ribosome from Candida albicans. So we also studied pathogenic fungi. And interestingly, so when we compare the mechanism of drugs against bacteria and against fungi, they're very different. So they're very different classes. I mean, there are some similarities, of course, of the drugs which, for example, target DNA RNA synthesis in both cases. And in fungi, normally most of medications actually target the cell wall function. So you may, let's say, weaken the cell wall and cells will die eventually, or pierce it, etc., etc. Or you can actually, well, interfere with ergosterol synthesis. But what we noticed and what surprised us a lot, that many drugs in bacterial world, they actually target ribosome. Because, I mean, if you stop the synthesis of proteins in the cell, well, eventually it will die. So it cannot continue without, without proteins. And, well, of course, you may say, okay, there is a problem of cross-reactivity since a fungal ribosome is eukaryotic, so clearly it's very similar to one found in us and humans. So you may expect that it's very hard to develop a specific drug, but nevertheless, we thought, okay, it's better actually to solve the structure and understand if we can find some differences there. Because again, based on bioinformatics, there are certain proteins in ribosome which, of course, diverse. They are not 100% identical, and there are even cases where certain drugs do not work on Candida, for example. So we were interested in the structure. So we published this work beginning of this year. And again, so some of this work has been done using high-end Titan cryos and K-free, but some of the work also was done using in-house Talos Arctica. Clearly here you don't have a problem of contrast because this is few megadaltons structure, and we get resolutions between 2.3 to 2.8 for different structures. So this is just for the vacant ribosome, but we also solved several complexes with different drugs. So beautiful structure, gigantic, well, hard to build. So if we look at the local resolution, we can actually see that the large subunit is very well resolved, so up 2.1 angstrom or so. So the smaller subunit is less well resolved, but nevertheless, we use different tricks like focus refinement, etc., etc. So we managed to build almost complete structure. So to show you the quality of density, so there are several examples where you can actually clearly see alternative positions of side chains, which, well, normally you see in crystallography also resolution of 2 angstrom or so, and normally they are not observed in electron microscopy. But, well, we could clearly see that we have different rotamers present, different alternative positions of side chains. We actually could also see the modifications on the side chains And well, yeah, that's been a lot of work going through all these side chains and checking it for modifications. So I told you also about these differences in binding, and it was known from bioinformatics already that there's in certain protein L44, there is a substitution of proline to glutamine residue. And actually, it has a large impact on the binding of cyclohexamide. It's one of the known drugs because it's known to be bound. So it targets E site and it blocks the movement of ribosome along a messenger RNA and prevents the release of tRNA. So actually what we did, we solved the structure of cyclohexamide in really large excess. So normally it doesn't bind, and we really had to push it. And what we realized, actually, if a cyclohexamide were bound, like at normal concentration, known concentration, there is a serious clash over here with the side chain of this glutamine. So it's less than 2 angstroms, so there is a sterical hindrance. So only when you do a real excess of this drug, you start pushing the side chains away and it can somehow bind. So actually, position of this residue explains why albicans, Candida albicans, is resistant to cyclohexamide in the wild type. And that kind of information can be actually exploited later for the rational drug design. So that's why you need the structures, actually. So we also studied different drugs like phylantozid, for example, checked its position. So we'll not really stop here. Blastocidin. So these are all known drugs, well characterized, and we actually were just checking whether they bind in the same mode and is the binding pattern is similar or not. And it turned out to be, for these particular drugs, very similar. Same for anisomycin. So I'll not stop here for more details, but we'll just share some final thoughts with you. So if you have access to, well, 200 keV or even 100 keV microscope, but with a decent camera, you can actually do quite a lot. So most of the things you can characterize pretty well. With resolution of 3 angstrom or so in EM, you can actually conclude quite a lot. Yeah, clearly you will not see hydrogens or waters, but yeah, for many things, this is good enough. So if you have a very small target, let's say like a soluble protein or complex which is below 100 kilodalton, so try to increase the size. So there are different approaches like nanobodies, synthetic bodies, or you can really, if you can, get to this state-of-the-art microscopes with energy filters, and it works. It really works very well. So you may have heard of faceplate, but there are so many problems with faceplate that it never became a mainstream. But this new generation of energy filters, they work beautifully. And importantly, quality of your sample is actually the most crucial thing. So if your sample is good, well, you will get the good data. If sample is crap, well, it doesn't matter what kind of microscope you're going to use, it will be still poor data, hard to analyze. So there are plenty of people involved in this project. I will not name them all and will not name all the funding, but I want to thank you for your attention. And if you have any burning question, feel free to ask it now or drop me an email later. Thank you.
Thank you very much for your lecture. I think we have time for only a few questions. While preparing, I have a question. I was very excited to hear about the heterogeneity of the samples. So can you please tell a bit more about those alternative conformations of side chains you mentioned? So how you deal with this, and is there a way how you can select the conformations early in experiment?
Yeah, well, so this kind of heterogeneity, let's say, is on very low level of a side chain. So this you cannot really check for or select for. You can only model it. So let's say when you have different conformational states, the position of the whole domain differs, or the orientation of domains against each other is different. This is normally you can pick up already at the level of 2D classification even, or 3D. So you can really generate 2 different volumes, 3 different volumes. And algorithms now so good that they can actually do this job for you. But you need decent amount of particles. So the more particles you have, yeah, the better it's gonna work.
Thank you. Unfortunately, we don't have time for questions and we have to go on. So thank you very much once again for the lecture.
Yeah, bye-bye everyone. I'll just stop the share.
And our next lecture will be from Yury Rizhikov. He's here in the audience, so please welcome. He's from Moscow Institute of Physics and Technology, and he will give us a presentation about the possibilities of small angle scattering techniques and the analysis of soluble membrane proteins. Please.
So, yeah, I will talk about ambiguities and completeness of analysis of membrane proteins using small angle scattering. And I have divided my presentation into 2 parts. In first part, I will give some introduction to just what small angle scattering is. And in second part, I will talk about our experience of investigation of structure of membrane proteins using small angle X-ray and neutron scattering, about problems and ambiguities which we met while investigation. So let's start. Small angle scattering is a low resolution method. The advantage of this method is that to investigate your object of study, you are not require something like protein crystallization procedure, which you need in case of X-ray diffraction, for example. And you can research your protein in native environment. And there are 2 types of X-ray scattering that are used. They are small angle X-ray scattering and neutron scattering. That are complementary methods, and they are powerful when we use them together. Sometimes it opens a lot of possibilities to understand our objects. So some formulas. First of all, how experiment scheme usually looks. There is a direct beam, X-ray or neutron beam. And there is a sample which scatters this radiation, and we measure this scattered radiation using a detector, and then analyze this intensity of the scattered radiation, and we are trying to analyze the sample, what the structure of the object that we study. The amplitude of scattered radiation is an integral which is written on the slide. All we know, this is the Fourier transform from contrast of density. This is not mass density, this is the density of scattering lengths. In case of X-rays, this density is proportional to electron density, and in case of neutron scattering, this is non-intuitive value. So in experiment, we cannot measure amplitude, we can measure only intensity, which is the square of modulus of amplitude, and we lose information, and the specific of small angle scattering is that we have ensemble of particles that are randomly oriented, and we also lose information because of because of this angular averaging. The problem that we try to solve is to find our function of scattering length density distribution from intensity function. And there are two difficulties. First of all, this is the phase problem, the same as we have in X-ray diffraction, and also angular averaging. So as I said, there is density distribution, and Fourier transform gives us amplitude function. This is reversible procedure, but when we measure average intensity, the only way to obtain this raw function is modeling. So we can build some models of our object and minimize the difference between experimental data and data corresponded to our model. Yeah, in general, there is uncertainty principle for small angles. Intensity at small angles corresponds to shape of the object of study. The middle range of scattering vectors or angles can open some information about overall fold of protein or some other object. And the biggest scattering vectors correspond to atomic structure. So let's start to the second part. This is the small angle scattering for membrane protein research. Membrane proteins are amphiphilic molecules. They have hydrophobic and hydrophilic parts, and they cannot be just in water solution. We require for membrane proteins some membrane mimicking systems, and when we investigate structure of membrane proteins, we have to take into account the membrane mimicking environment that we use, and if we don't do this, we can obtain some artifacts. So our object of study is the sensory rhodopsin 2 transducer complex. This is the photosensory. This complex is responsible for negative phototaxis in Archaea, Notronomonis pharaonis. And when we investigated this protein, we firstly tried to understand which oligomeric state it has. Theoretically, we know that, first of all, this has to be dimer, and also this type of proteins have to form trimers of dimers, and this slide about the contrasts of different parts of the protein detergent complex. The specific of detergents and lipids and many of amphiphilic molecules is that the hydrophobic part of the systems usually has electron density which is smaller than electron density of water, and the contrast is negative. And because of the presence of this part with negative contrast, we usually have a local maximum at the scattering profile. And the first mistake that you can obtain when you research your membrane protein without taking into account detergent belt is that you will have some correlation distances which correspond to this correlation lengths, but in your protein they are not presented. And when we investigate our complex of sensory rhodopsin with transducer, first thing was that, okay, we see trimers of dimers or shaped trimers of dimers, but of course it is not true because we used for our model of equal density object. And for small angle X-ray and neutron scattering, it is really important to use as more a priori information as possible for your object. For example, in our case, we can build some molecular model of our object, and what we can obtain when we use when we have molecular model, we can add pseudo-atomic pseudo-atoms that correspond to detergent belt, and we see that the dimer model is good for describing our X-ray scattering data. And this is a correct way to treat this data, because if we don't take into account detergent belt with their negative and positive contrasts, we will have this artifact presented as correlation lengths in our structures. The advantage of neutron scattering is a possibility to use heavy water or mixes of light water and heavy water, and these two waters has different scattering length density. And for example, when we use clear D2O heavy water, we have all contrasts are with the same sign. And in this case, this is not so big problem if we use models with equal density. And when we use models of equal density, which are usually used in ab initio algorithms, we can obtain structures that are similar to the true structure. This is just a comparison of Sachs and Sons results. And the next ambiguity in analysis of small angle scattering data is oligomerization polydispersity of our proteins or other macromolecules that we study. So the complex of sensor rhodopsin with transducer is from Natronomonis pharaonis, that is halophilic archaea. So the salt concentration in the buffer of the protein is important, and when we prepare several concentrations of the salt, we obtained different scattering curves. At low salt we have dimers, but in high salt we have the part of trimers of dimers. Trimer of dimers is more native oligomeric state, and we obtained it in more native buffer. And if we take into account the possibility to have a mixture of different oligomeric state, we can take into account and obtain correct structure of oligomer. For example, here there was a mixture of monomers and trimers, where monomer is a dimer of sensory rhodopsin transducer complex. And this fitting by two or more compounds is more precise when we use several curves with a different proportion of these compounds. This structure is it corresponds to tripod-shaped trimers of dimers, which we investigate in more details and build the model. And there were two possible types of trimer of dimers. This is like with contacting transmembrane part or not contacted, and we show that this tripod-shaped model is correct because mixture of dimers and trimers of dimers approximated the experimental data better. But if we, for example, use only data for the highest concentration of the salt which we use in our experiments, and if we will approximate this data by the monodispersed model, we can obtain some result which also looks reasonable, because there were also models Y-shaped and O-shaped, which are not like this model. In this model, we have contact only in the tip of cytoplasmic tip of the protein, and there we have contact through the all lengths of the protein, and we obtained structure that is in good agreement with this model and also good approximated the data. But this is an artifact which we met because we don't take into account oligomerization polydispersity. So in study of membrane proteins, there are two main things which you have to take into account. This is the detergent belt, or for example, if you use nanodiscs or some other membrane mimicking system, you should take into account their presence. And the other thing is the possible oligomerization polydispersity, which can take place not only in membrane proteins, but also in case of soluble proteins. That's all. Thank you for attention.
Thank you. So we have plenty of time for discussion, but also I think we can go on with discussion during the coffee break. So anyway, please, questions for our lecturer.
Thanks for the talk. So my question is about the membrane mimetics, which you have on your samples. How do you take into account the free particles? If you have micelles, you will always have free micelles, which will spoil your curve, in my opinion. So how do you handle it?
Yeah, the best way is to use the sample from gel filtration when your buffer has some free micelles and also your peak fraction of your membrane protein has the same concentration of free micelles, and when you subtract your background buffer, you will have just clear curve only for your protein with detergent belt, but without contribution of micelles.
I have a question. What additional methods do you use to validate or refine your model? So how you can discriminate between O and Y structures using additional methods?
The best situation when you have for your proteins some molecular model or molecular models of different domains, and in this case you use your data just to combine their orientations and positions. So yeah, in this case, the ambiguity is not so big. Also, of course, you can use gel filtration and other methods to estimate your approximated molecular weight, and the multiplicity of the structures that you can obtain have to correspond to the calculated molecular weight, for example.
Okay, thank you.
Additionally, you can use cross-linking with SDS page to validate your oligomeric states and many other.
Okay, thank you very much. This was really interesting. I actually never seen such big proteins studied by SACS. So this was really exciting. Thank you. So our next speaker is Professor Jörg Pieper from University of Tartu. He is very well known among the photosynthesis research community for the application of complex and sophisticated physical approaches to study biological samples. So, Professor Pieper, please start when you're ready.
Yeah, thank you very much for the nice introduction, and good morning to Moscow, in this case from Germany. You have already received a very good introduction into small angle scattering methods in the previous talk, so I can probably just add another perspective from my side and want to show you, after a brief introduction into small angle scattering, some examples that may motivate further work using small angle scattering. This includes examples concerning studies of oligomerization of photosynthetic complexes, complex formation between different protein complexes, the study of membrane proteins and their detergent shells, pretty similar to what we have just seen. And then finally, some light-induced structures or conformational changes that were induced by external triggers. Should I have time, I can say a few words about molecular dynamics studied by neutron spectroscopy. Well, let's start with a brief introduction to small angle scattering. There's quite some chance that you have seen small angle scattering even in your daily life. Here you can see a photo that I have taken on approach of Tallinn Airport from an airplane. And this shows you essentially the very basics of small angle scattering. What you need is a source of a particle wave that you can use as a probe for the object that is under study and that scatters your particle waves into different angles. And finally, you need something to monitor in this very basic example here. These were the clouds above Tallinn. And the very basic condition for observing small angle scattering is that the wavelength of the probing particle waves are much smaller than the dimensions of your object. So, of course, in my talk, I want to solve the question, what do we have to do if the object of our interest is a biomolecule? Now, why should you use small angle scattering at all, especially that it's a low resolution technique, and you have seen earlier this morning very nice examples of high resolution structures obtained by different methods. So the arguments are the following. You can basically study shape and size of biomolecules in solution, and you have no need for crystallization, staining, or cooling, so you can work under nearly physiological conditions. That means you can prepare a direct link between the structure of an object and its function, which is often studied by experiments, for instance, by spectroscopy in solution. And to be more precise, as I said in the outline of my talk, we can check whether samples studied by spectroscopy are affected by oligomerization or aggregation. In the simplest case, we can study complex formation, and we can study detergent belts, micelles, vesicles, and lipid bilayers, which are inherently heterogeneous and not so well approachable by high resolution techniques. And finally, a major challenge is to study protein conformational changes that are induced by external triggers. And as I said, the final goal or the highest potential of small angle scattering is if you can link the structures obtained by small angle scattering to experiments on their function. And this is fortunately in the recent years aided by very powerful new simulation tools like ATSAS or Pepsi simulation software, and I will show you examples of this a bit later. So still, let's start from the question, why do we observe small angle scattering, and what characterizes the interaction between neutron and atoms in the sample? And the basic parameter is the so-called scattering length B. And as you have already heard, for X-rays, they are scattered by the electrons, so that the scattering length there is proportional to the atomic numbers and rises linearly with the atomic number. Neutrons, small angle scattering is complementary nicely to X-ray small angle scattering, because neutrons are scattered by the nucleus. So completely different scattering lengths apply. And especially you see some examples here at the bottom of my slide. you can see that you have very different scattering lengths, even negative and positive, for different isotopes, here in this example for hydrogen and deuterium. And this gives us the chance to exploit so-called contrast variation in neutron small angle scattering in contrast to X-ray small angle scattering, because if you replace hydrogens by deuterons, you have a completely different contrast to your solvent. Another parameter that plays a role, but I don't want to go into details here, is the scattering length density, which is essentially the scattering length per volume. And this is calculated on the slide here for H2O and for heavy water D2O. And again, because we are working with hydrogen and deuterons, the scattering length densities become negative and positive respectively. And this is the basics of doing contrast variation and contrast matching, which is shown in this slide here, where the scattering length density of water mixtures are shown as a function of percentage of D2O in H2O D2O mixtures. This is this linearly rising dashed curve. And if you compare that to the scattering length densities of different biomolecules like surfactants, like lipids or detergents, you see that there are crossing points. These become the so-called match points, which means if I can tune continuously the contrast of my solvent, then I find points where the contrast of the solvent is more or less the same as that of my detergents in my sample solution, or of protonated proteins. It's a little bit above 40% of D2O in the solvent, and D proteins are best approached or matched around 100%. And this gives you this additional possibility to switch on and off the scattering of certain components in your sample mixture, even if you have a complicated solubilized membrane protein in buffer solution. And this slide just gives another illustration of the same fact, where, for instance, you have a protein shown in blue here, surrounded by a detergent belt in yellow. And if the color of, which means the contrast of your solvent, is rather similar to the protein, you highlight the scattering from the detergent belt. And if you move on, changing the contrast linearly of your solvent, and you see that you find a match point for the detergent belt as well. And later on, I would like to show you a few examples where we exploit this. And here you have seen a similar slide already of the basic setup of a small angle scattering instrument with a source, in this case, of course, for neutrons, which can either be a reactor or a spallation source. For you, the closest would be the research reactor in Dubna. And then the source sends probing neutrons to the sample, where they are scattered and detected on a two-dimensional detector to measure the small angle scattering. And finally, we plot the scattered intensity as a function of the so-called scattering vector, mostly shown as Q, sometimes as S, which is a function of the scattering angle theta and depends on the wavelength of the probing neutrons. And to complete the circle, you might see the similarity of my simple cartoon from the first slide of this. So to complete the introduction, we have a master equation that describes small angle scattering, which is the intensity as a function of the scattering vector. And the main parameters here are this delta parameter, which gives the difference of the scattering length density between particle and solvent. So this is the parameter that tells you, have you contrast matched a component in your sample, or do you have a contrast which gives you a nice scattering from this component? Then you have a so-called structure factor, which in dilute solutions of proteins tends to be 1. So you don't have to consider it. But for instance, if you have membrane stacks where you can define a unit cell which is repeated within the stack, then this structure factor becomes interesting and has to be taken into account. And the most important is the particle form factor, which gives you the shape and size of the protein under study. Now, for analysis, a few words. The simplest model-independent analysis is the so-called Guinea approximation, which you can apply under this condition where the value, the plane value of the scattering vector times the radius of gyration of your sample is smaller than 1.3. So basically you find that in the limit of small scattering angles, but it always depends on the size of your protein. And within this range, the scattered intensity becomes exponential or depends exponentially on the scattering vector value and the radius of gyration. So if you plot this value as on a log scale, you should find a linear range, provided that you have a monodispersed sample, which means that within your sample solution, there should only be one type of protein. Well, this shows you some model calculations that we have done for illustration of small angle scattering, where we have used one and the same protein monomer and formed different types of dimers, extended dimers and compact dimers and trimers from one and the same monomer. And the important thing here is that, as you have in principle also heard in the previous talk, the intensity of your scattering depends on the pair distances that are present in the sample, and they are related by Fourier transform over all distances from 0 to the maximum distance that can occur in your sample. So from the scattering data, you can derive via Fourier transform the distribution of pair distances present in your sample, and these pair distances characterize your sample structure. But now let's look at a few model calculations from these very simple protein assemblies here. And the theoretical curves that you expect in small angle scattering from these 4 different molecules you can see here. The black curve is the monomer. And you see that this curve is the one that is shifted to the highest scattering vectors. And here you see the inverse relation between scattering vector Q and the distances that you find in your sample. The smaller these distances on average, the higher the Qs where you find your scattering curve. And in opposite direction, the largest object that we study is the trimer, and this curve is shifted to smaller Q. So you see the inverse nature of scattering vector and distance in real space. More interesting then are the pair distribution functions. You see the black curve for the monomer, which as we expect, gives you the smallest radii in your structure. And for the compact dimer and trimer, this curve is shifted towards higher radii, as you would expect. Most interesting is for us the long dimer cylinder, because that one, of course, has the longest possible pair distance function at all, so it extends to the highest value of radii here. And this gives you an impression how small angle curves look like, and how by Fourier transformation you can obtain the distribution of pair distances characterizing your structure. So that brings me to the second part of the talk, which summarizes some examples about simple studies of oligomerization, complex formation, membrane proteins, and conformational changes in proteins. And the systems that we study are phycocyanin and assemblies thereof, the photosystem 2, very famous photosynthetic enzyme splitting water into protons and oxygen, and the orange carotenoid protein. And the processes we want to see here are oligomerization or complex formation for the phycocyanin. For photosystem 1 and 2, I want to demonstrate some effects on membrane proteins, and finally conformational changes for OCP. And again, the message that I want to bring across is that we deliver structures in buffer solution and can nicely correlate high resolution structures with experiments on function. And so let's start with oligomerization. We got a sample called phycobilly protein of the special cyanobacterium Acariochloris marina, which should be a rod-shaped structure. But our colleagues found the following time-resolved fluorescence curves here. If they had their phycobilly proteins in a buffer with phosphate, there was always energy transfer to about 675 nanometers, while in a phosphate-less buffer, this energy transfer switched off and fluorescence originated from higher energy. So the question was, why is this energy transfer impaired? And you can solve that with very easy methods. First, we started with solution measurements of phycocyanin only. And in high-resolution structures, you find hexamers and trimers of them. And if we compare to our curve, you see that in our solution, we found trimers. So that's a very simple check of oligomerization state of a protein. But we were indeed interested in the structure of this supposedly rod-shaped sample in phosphate-containing and phosphate-free buffers. So we found a very nice small angle scattering curve for the phosphate-containing sample, while this structure was almost completely lost in the phosphate-free sample. But in addition, you see that here is a strong increase of scattering signal towards small q. And as I said, small q means large structures. So we see we see a strong aggregation in addition to this almost lost structure here. And we could fit both curves with very simple geometrical models of cylinders, because we intended to find a cylindric structure here, and the phosphate-containing sample had a cylinder length of 225 angstrom and a radius of 50 angstrom, while the phosphate-free sample had only a length of 28 angstrom left. So we could conclude that in the phosphate-free buffer, the samples are decomposed, and could find a structural reason why the energy transfer in this sample was impaired. And that for us is a very nice example how with very simple geometrical models you can explain data from functional measurements in solution by small angle scattering data on almost the same sample in solution, where we can derive the structure of PC primers for the phosphate free sample, and we can derive a structure of 8 phycocyanine and allophycocyanine subunits for the phosphate containing sample. Now, a few words about membrane proteins, and of course we also ran into the difficulty that in solution they are surrounded by a detergent belt. I remind you on this Gini approximation that we should find a linear range in low Q. And the first example is LHC2, where we studied 3 different protein concentrations. And you nicely see that only the lowest concentration is linear and free of aggregation, while all the higher concentrations show a strong aggregation here. So we should warn our colleagues, our spectroscopists, to do experiments on the aggregated sample and limit them to only the lowest concentration. And here you see the structural model that we derived for LHC2. Now, the photosystem 2 measurements, where we found in 5% D2O a very nice small angle scattering or structure obtained by small angle scattering that fitted the high resolution chimeric structure very well. And that's at first glance a surprise, because we say it's a membrane protein. You have to have a detergent belt. But the reason is that we were at the contrast match point of the detergent almost. So we are about here, and to see the full protein with the detergent belt, we can do a small angle experiment in 100% D2O or a SAX experiment, and then you can make a model for the detergent belt and you find a monomolecular layer of detergent molecules. However, I have to point out that, as the previous speaker did, these contrast matching experiments are extremely difficult, because for the detergent head groups and tails, you see a similar effect as for H2O and E2O. The scattering length densities are very different and require different match points in principle. And what is often done is to just apply the average match point. And what can happen to you is, I show you here some data of photosystem 1, where we used specifically butyrated detergent, which has a similar scattering length density over the whole molecule. And you see that for the same protein in different detergents, you find seemingly different sizes. And the reason for that is that simply protonated detergents cannot be perfectly matched because of this heterogeneity of scattering length densities. But if you employ specifically butyrated detergents, then you also find a different ab initio structure, which is quite a bit smaller than the unperfectly matched structure of the same protein in protonated. So let me go over, because I'm running out of time a bit, and show you the last example here. That is a very nice collaboration with colleagues at Technical University Berlin and Evgeny Maximov at in Moscow, where we studied the structure of OCP in the compact ground state and by in situ illumination in the active state. And for that, we had to build a little setup where the sample could be illuminated in the SUNS machine. And you see here the sample that was illuminated turned from orange to dark red, indicating that we really found or have achieved the active state of OCP. And then you see that the small angle curves here become quite a bit different. And we have used then modeling tools and could model the dark measurement with a dimeric high resolution structure. But fortunately, we now have very nice simulation tools which allow you to define rigid and flexible structural parts, the flexible ones indicated here by these dummy molecules. And you can see that this dimer has extended quite a bit by separating the two domains that OCP has and by unfolding the N-terminal extension. And with that, we find a very nice model for this active state OCPR. So actually, in the very last part, I just wanted to make you aware that structures are not rigid and that this can also be studied by neutrons. So I show you an MD simulation by Dr. Tobias, where you see in dark gray the protein wiggling, and on the surface you see the water molecules. So this is just shown to illustrate that we have to keep in mind our proteins are dynamic entities and they are moving on very different time scales, starting from femto and picosecond time scales for small side chains. And here you see a temperature dependence of that mean square displacement of the photosystem 2 as a function of temperature. And with this, I want to conclude and thank all my co-workers for the Photosystem 2 project, mainly Maxime Golob, my postdoc in Tartu, but our co-workers in Germany, France, and Poland. And phycobilirubin protein project was also with German colleagues, but measurements done in Dubna with Sasha Kuklin. And OCP, of course, together with Yevgeny Maximov and the colleagues at TU Berlin. And I thank you for your attention.
Thank you, Professor Piper. We have time for some questions. Please, Konstantin.
Okay, Jörg, thank you so much for a very interesting talk. I have a very general question. What is the minimum size of the object you can study with this technique? I mean, for example, can you study the interaction of a certain protein with antibody Fab fragment, for example?
Oh, I'm not familiar with that protein and antibodies, so we could probably talk personally. In our work, we can go to sizes down to, let's say, 10 angstrom, but it also depends on the instrument, of course. There are small angle and ultra small angle machines, but the race is more for larger objects in that case.
Okay, thanks.
So, dear colleagues, it is 2 o'clock, so it is time to continue with our second session. And the first talk of the second session is from Olga Sokolova. Professor Olga Sokolova is probably the major specialist in cryo-EM in Russia. Yeah, Olga, nice to see you. Olga is now in China, therefore she is participating online. So Olga, the floor is yours. You're welcome.
Okay, thank you very much. I hope that you hear me well. So thank you for naming me the greatest person, I mean, the main person of cryo-electron microscopy in Russia, but I don't think I'm the only one cryo-electron person in Russia, because right now, hopefully, we have more scientists and more labs in Moscow, and not only in Moscow, St. Petersburg, in other cities who are also involved in cryo-electron microscopy. So I'm thankful for the organizers to allowing me to speak at your satellite conference. And today I would like to talk about chaperones. So what are chaperones? If you imagine the chaperones in human world, then the chaperones are people who usually help other people, for example, young kids to, here is, for example, chaperone is assisting kids to enter the school bus. Exactly the same molecular chaperones are, they're doing the same. They help young proteins and they aid in folding of newly synthesized polypeptide chains and translocation of proteins across the membranes. Chaperones are usually released after proper protein folding. So there are several families of chaperones that are known. And there are chaperones and also chaperonins. Chaperonins are one of the most studied families of chaperones. They are multimeric complexes that ensure the proper folding of newly synthesized polypeptide chain and also prevent aggregation of denatured proteins in an ATP-dependent manner. So chaperonins are usually, so far, they have been divided into two groups according to the structure and origin. And here I present these two groups. Group 1 accommodates bacterial, mitochondrial, and chloroplast chaperonins, and with the most studied representative to be GROEL from E. coli. So these chaperonins, they are arranged as two back-to-back rings, with each ring containing 7 subunits, and each subunit comprised of 3 distinct domains. Equatorial domain form the interface between the 2 GROEL rings and also contain the sites of nucleotide binding. And it also provides the inter-ring communications. Then there is the apical domain, which are responsible for closing the cavity where the protein folding occurs. And then there is a group 2 chaperonins. It accommodates eukaryotic and archaeal chaperonins. And member of this chaperone family, they have 8 or 9-fold symmetry. And also they form back-to-back rings in homo or hetero-oligomeric complexes with the individual subunits retaining the same domain structure with a high degree of sequence identity and or similarity to GROEL. So most studied group 2 chaperonins are eukaryotic trig and also archaeal thermosome. So the most difference between group 1 and 2 is a number of subunit, and also that group 2 chaperonins, they lack the co-chaperonin. A co-chaperonin is a special protein which helps to close the cavity. So here cavity in GROEL is called with this GRO-ES protein, and this is a co-chaperonin. So there is an ATP binding site in the equatorial domain of each ring, and GROEL drives its protein folding cycle by cooperatively binding ATP in one of 2 rings, priming that ring to become folding active upon GRO-ES binding. So GRO-ES on the top. And then there are 2 structures of GROEL with bound GRO-ES. One is a football shaped, which has both GRO-ES on top of GROEL, and then a bullet shape, which has only one GRO-ES in contact with the upper ring. So here, in this table are listed all structures that have some ligands in one or both rings. So most of them, it's ATP in cis ring, which is top ring, and also magnesium, or ADP or ATP in trans ring, which is on the bottom. But there is no structures deposited to PDB in which ADP is in both rings. And here we decided to fill this gap and solve the structure of GRO-EL, GRO-ES in presence of ADP. We use cryo-EM for that, and you can see that there is a complexes only in bullet form when the ADP is bound. So here the resolution obtained were 3.4 angstroms, and we found two major structures with different orientations of apical domain in the trans ring. So these, in the right structure, apical domains are located much farther away from the symmetry axis comparing to this A figure. So we denoted them as a wide structure and a tight structure. And then when we look at the intermediate domains of the trans ring, then we can notice that actually those domains are responsible for this conformational change. So we evaluated this scattering density for the nucleotides in the pocket, and we decided that for both ADP conformations, the binding pocket is much better seen than the region of bound nucleotide. So by the mutual arrangement of these amino acids, we can draw an indirect conclusion that as to which nucleotide, ATP or ADP, are the bound amino acids. And this is our structure, is the first structure in which is in a bullet shape, and has bound ADP in both rings. And then recently, about 5 years ago, the new class of chaperonin was discovered. It is called viral chaperonin. And these viral chaperonins are coded mostly by giant bacteriophage genome. So here is the first three-dimensional structure of viral chaperonin from bacteriophage EL, and it was published in 2016. So giant bacteriophages, they are viruses that infect bacterial cells, and generally bacteriophages are extremely diverse in size, morphology, and genomic organizations. And then here you can see that FICA-Z phage, which is giant bacteriophage, and T4 phage, it's a normal bacteriophage. So the giant bacteriophage, if you compare it to the T4, it's much more, it's larger. It's about 3 times larger and taller. So that's why it has been named as a giant bacteriophage. These giant bacteriophages, they were discovered back in 1980s in Moscow lab. So, and here now there's actually several of these giant bacteriophages known. So FICA-Z is the first discovered giant bacteriophages, but now it's also OBP, AL201, Phe201, and PA3, and so on. And actually the number of giant bacteriophages, it increases now. But if we can see here, there are the genomes of all these known bacteriophages. They are aligned. And if you can see that here in orange are the arrows, and they pointing to the reading frames of structural proteins of the phage, for example, of OBP phage, which is first in this row. And this orange arrows, they pointing to the reading frames of head and tail. But then in blue are the extra structural special proteins, which are designed to help phage to survive inside the host cells. So unlike some smaller viruses, giant phages, they come into the host cell fully equipped with their own expression machinery. So they are expressing its own RNA polymerase, helicase, and also chaperonin. So here is the chaperonin gene product 246. So if we compare the domains of chaperonins, so here is the equatorial, intermediate, apical domain, then we can see that viral chaperonins, they have similarity with both chaperonin groups, group 1 and group 2. Like group 1, they have 7 subunits and they are organized as a barrel structure, not only double, just a barrel structure. But then when, like group 2, they don't require the co-chaperonin to close their chamber. So, and they do not have this insertion in apical domain, which is, so they do have this insertion in apical domain, which is required for group 1, and they do not have this helical protrusion like in group 2. So according to sequence analysis, phage chaperonins, they seem to be closer to group 1 chaperonins. And this evolutionary proximity of phage chaperonins to group 1 chaperonins is also indirectly indicated by their location on the phylogenetic tree. So we, first what we did, we solved the three-dimensional structure of OBP giant bacteriophage, GP246 chaperonin. And for the first time, we found that it actually is organized not as a double-decked barrel, but as a single ring. And also it is protecting only small proteins. So here we can see the data of isothermal titration calorimetry and with the stoichiometry values for titration with the ATP and ADP. And also on the right-hand side, you can see the results of dynamic light scattering. So for example, double ring chaperonins EL, which is here shown in green, it can protect both proteins, large like GPDH and small like endolysin. But if you go to the larger proteins with the OBP chaperonin, then you can see that for GPDH, OBP chaperonin do not provide any protection. So it protects only the small proteins. So we've been thinking how the single ring chaperonins without co-chaperonin can protect the proteins. So we decided to solve the three-dimensional structure of it, and we use cryo-EM for that. And here, both Dr. Tatiana Stanishneva-Konovalova and also our Chinese master student, Van Si, they both working on this three-dimensional structure. And you can see that this structure of bacteriophage encoded chaperonin is a single ring of 7 subunits. So this already distinguishes it from most chaperonins, which are usually consist of 2 rings. And then also, very interesting thing is that this chaperonin is assembled from 7 chemically identical subunits, but it only displays this C7 symmetry at the level of equatorial domains. And then apical domains are arranged completely differently. They are arranged as 3 pairs. You can see here 1 pair, another pair, and 3rd pair. And then 1 unpaired domain, which is actually, its density is blurred because it looks like it's in a continuous motion. So this raises the questions, how this role of this free subunits, so we call this A and B, and then this free subunit we call C. What is the role of this free subunit? And is it possible to switch the roles A and B and C? So we did also some molecular modeling. We performed, with the help of Dr. Pavel Semenyuk, we performed the homology modeling for both subunits A and B. A here is in red, blue is in cyan, and then the alignment indicates the preservation of the nucleotide binding site in both domains, and also the hinge residues between the equatorial and intermediate domains in this OBP chaperonin. So with the inter-ring interface, the situation is completely different. In the GRO-EL subunits, two sites are responsible for inter-ring interactions. And they both, first site is lysine, alanine, and the second are arginine and glutamine and valine, and both sites are not conserved in the GP246 sequence, as you can see here. So we compare the atomic models of A and B subunits to the structures of GRO-EL subunit in two conformational states. And then we can see that the bottom hinge here is present in OBP chaperonin because it's likely providing the rotation of the apical intermediate part relative to the equatorial domain. And then the most remarkable difference in the structures are observed in the apical domains. They caused by replacement of amino acids in the top hinge of OBP chaperonin, and then corresponding glycine residues are replaced by large polar residues whose phi and psi values of torsion angles cannot provide efficient cis-trans switching. So here we can see that the upper hinge in OBP chaperonin is broken. That's how we call it. So, and then conformations of, if we compare conformations with, of ABP A subunit with ABP B subunit, we can see that they actually are quite different. So if we compare them with the different states of GRO-EL, then we seen that A subunit of OBP chaperonin is somewhat similar to the conformation of T state of GRO-EL, which has low affinity to nucleotides. And B subunit conformation, which is cyan here, cyan, it's more similar to ATP-bound conformation state of GRO-EL. So unlike in GRO-EL, here in the nucleotide-free state of OBP chaperonins, both conformations are represented simultaneously. So then we investigated the interaction of OBP chaperonin with different nucleotides. And here Evgeny Pichkur helped us a lot. So we solved two asymmetrical 3D reconstructions of ATP gamma S bound form and ATP gamma S. It's a non-hydrolyzed analog of ATP, and also ADP bound form. So here we can see that actually both reconstructions have even more heterogeneity, and less subunits can be resolved. So it looks like now the more subunits, upon binding the nucleotides, more subunits became C-like. And here we can see that, for example, in the three-dimensional structure of EL chaperonin, there used to be, they claimed that they solved two closed single ring conformations, which later united into one big closed double-decker. But recently it appeared that this is not true. So we found, when we studied our nucleotide binding, we found several conformations of the subunits. And we found that 50% have only one subunit which was not resolved, 30% have 3 subunits which were not resolved, and 20% have only one pair, N and B, and all other subunits were in these unstructured C conformations. So here you can see this is a conformational mobility of ATP gamma S bound chaperonin, we can see that there's a large conformational transitions of at least 5 subunits. And then one of them is clearly changing its position between A and B. You can see it goes from here to here. It's huge conformational change. So, and since the top hinge in OBP chaperonin is not functional properly, so probably these conformational changes may only be provided by rotation of the bottom hinge. So then we studied these intersubunit contacts using molecular modeling, and we see that in ATP gamma S bound conformation, there is a formation of alternate sides of intersubunit salt bridges, and also some of the intersubunit salt bridges here are breaking. So for example, you can see here this B and A subunit bridge is missing, and also A and B subunit bridge, before they have 2, and now it's only 1 remain. So here we decided that this is probably the proposed mechanism of functioning of the single ring chaperonin. So we suggested the existence of 2 sets of nucleotide binding site in chaperonin subunits, which differ in binding constants and their corresponding heat effect. So you see here again this colorimeter titration. So you can see that this is the first site and this is the second site. So about 4 are the second site, and about 3 for ATP gamma S are in the first site. So we propose that these results reflect differences in the ability of A and B subunits to bind nucleotides. So here apparently so this is an upper form without any nucleotide, then ATP comes, and then first A subunit, which is has low affinity to nucleotides, and then B subunit has a high affinity to the nucleotides. So first, ATP may bind to B subunits, then it may bind to some A subunits, and then finally, we're getting this primed for substrate binding chaperonin, where at least 5 of the subunits are in C conformation. Then misfolded substrate will bind it, and if it is a small substrate, then hydrolysis of ADP will occur, and then finally the folded substrate will be released. But if there's a large substrate, then it may either partially bind to this chaperonin, or it may not, like, just bind and then be released without folding. So, and then these changing from this state to this large state subunit, probably it's just a one-way conformational change. So after the large subunit, after large substrate will bind, then after that it won't function properly. So then we studied the interaction of OBP chaperonin with a substrate using the cryo-EM. So here the images are obtained on GEOL cryo-ARM 300 microscope in GEOL headquarters. So you can see that even on 2-dimensional averages of the chaperonin, we see some extra density, which probably corresponds to the bound chaperonin. So we did this 3-dimensional classification with the help of Andrei Moisenko from Moscow University, and we found that some of these extra densities are located either in the hole between 7 subunits, or also they are interacting with the apical domains. So here we don't have a very high resolution, but even if you compare with the upper form on the right, the substrate bound form, it clearly shows an additional density close to apical domain in C subunit. And in general, these substrate bound form has more compact appearance comparing to the upper form. So we assume that substrate binding leads to compactization of these chaperone structures. So that's why we think that chaperoning OBP cannot effectively protect large oligomeric proteins. They just do not fit into chaperoning cavity, and they cannot lead to compactization. So we also did some molecular docking using rigid body docking to model the interactions of chaperoning with the substrate protein. And here we can see that again, it binds to one special subunit, which is not in this. So there's two subunits, another pair of subunits, another pair of subunits. So it binds somewhere between this B and C subunits. So sorry, they are here in this image shown just in A, B, C, D, E, F, G, but basically this is C and this is B. And that's why we think it's a differences, why the differences between conformation of B and A subunits. And this work was done by Tatiana Stanishneva-Konovalova as well. And then now, if I have some time, I can show you the very fresh work where we solve the structure of another chaperoning of giant bacteriophage IR9 from Bacillus subtilis. And this is another, it also consists of 7 subunits. So we got a pretty good structure with the resolution below 3.5 angstroms. And this was done by our master student, Ekaterina Maslova. So here you can see this is this C1 symmetry, and this is a C7 symmetry. So this chaperoning actually has more, it has less, all 7 subunits were resolved, and the local resolution map shows that the higher resolution is in the equatorial domains, very similar to OBP chaperoning, and less resolution is in apical domains. So we again did some full atom model of the heptamer. So this is OBP chaperoning, 2 subunits A and B, and this is a 2 neighboring chaperoning IR9 subunits. So here the main difference is the presence of this R202 and E402 ion pair in IR9. So in GROEL, this pair corresponds to residues R197E386, which plays important role in conformational rearrangement of GROEL. So in this sense, IR9 is even closer to GROEL comparing to OBP. And then we studied the conformational mobility. So
Olga, we cannot hear you. I think we should continue. Could you then write here that? Yes, anyway, let's thank Olga. I think we cannot imagine our current state of structural biology without NMR, even though, as we discussed during lunchtime, that the role of NMR is changing right now. but it still is a major player in this field, and therefore it is my pleasure to invite Konstantin Mineev to tell us about NMR. Konstantin is here, so he'll talk offline.
Does it work? Okay. Good afternoon, everyone.
Can you hear me now?
Olga, are you back?
Yes, I'm back.
Okay. I'm sorry, so you was frozen and we cannot hear the
Yes.
your the last slide.
Was it for long? I mean, was it a long time ago, or when did you stop hearing me?
I cannot tell you exactly, but that was the last minute of your official talk. So
Okay. So I'm actually done with my talk, right?
yeah, and unfortunately we don't have any time for questions. Yeah, we have uploaded you already.
Okay, thank you very much. So I
Thank
think
you.
if anyone wanted to ask me something, one can just write me the email.
Okay. Thank you.
Okay, thank you very much. Bye bye.
Bye bye.
So, shall I proceed?
Yes, please.
Okay. So good afternoon, everyone. I would like to start with thanking the organizers for giving me this opportunity to talk a little bit about my work. And I'm going to tell you a few things about NMR spectroscopy and how can we apply NMR to get the structures of membrane proteins. So I think most of you here understand that membrane proteins are important because they are prospective targets for drug design, and you can cure a lot of diseases, potentially cure by acting on membrane proteins. That's why we are interested in getting the structures of these objects. And I think also I do not need to persuade you that there are two major approaches to getting the structures of membrane proteins. Right now it's cryo-EM, which can routinely solve the structures of, say, ion channels, and X-ray, which can almost routinely solve the structures of G-protein coupled receptors and other proteins. But there are some proteins that do not surrender to cryo-EM and resist the crystallization. So I give here two examples. Okay. I give here two examples. The left example is a structure of platelet-derived growth factor receptor, which was obtained with the resolution of 2.7 nanometers. And the second structure is the toll-like receptor 5, which was obtained with the resolution of 2.6 nanometers. And these are the best structures available for the full-length proteins from this family. The family is called the single-pass cell receptors. So why they are called single-pass? They are called single-pass because they have very large extramembrane domains and intercellular and extracellular domains, but their transmembrane part is just a single alpha helix and nothing more. However, the erective S dimers or oligomers, so actually the real transmembrane domain is two helices, at least two helices, but it's very small. And that hinders the crystallization of the proteins, and they're quite mobile. And for this reason, they are not well studied by cryo-EM. So they're also quite important from the point of pharmacology because they recognize hormones, growth factors, pathogen-associated molecules, and etc. And they're involved in a variety of diseases, including cancer, neurodegeneration, inflammation, and so on and so on. So they are really used as drug targets. And since we are not able to solve the structure of the full-length proteins, we normally use this long ago proposed approach, which is called the divide and conquer approach. It was proposed for a completely different purpose, but we use it to study proteins. So what we do, we cut our proteins into pieces. These pieces should be functional or should fold by themselves without the other pieces. So we take these units, study their structures, and then try to combine the data and get the full picture. And when we apply this approach, this divide and conquer approach, we have some space left for NMR spectroscopy. So NMR spectroscopy is quite an exotic nowadays way to get the structure of the protein, but it's quite a potent approach for several reasons. So first of all, we study the proteins in a completely native environment, almost completely native environment. Yes, we can study structure and mobility all in one. So in the same experiment, we can understand the structure of the protein and which parts of the protein are mobile and which are not. And we can also investigate the disordered regions. It's the only way to study the disordered proteins or partial disordered proteins. And these single-pass receptors, they are quite rich with disordered domains. However, and that's the major problem with NMR, it's impossible to work with large proteins or large domains. So how does NMR work? So here on this slide, I show a typical NMR spectra. It looks like this, not like you're used to. This 1D line, so we have 3D something. And here's the NMR structure. So what is this 3D something? This 3D something is a 3D NMR spectrum which reveals the interactions between the nuclei in the molecule. The position of peaks, they correspond to the they help us to identify which exactly nuclei interact, and the intensity of peaks, they show the magnitude of the interaction. So analyzing this complex NMR spectra with some kind of mathematical approach, we can get the distances between the molecules and orientations of bonds and dihedral angles in our proteins. So we get structural data. And then we can convert the structural data to the structure, which looks like this. And this structure is the best model which corresponds to all the data that we have. If we make multiple calculations for our data, if they all converge into one kind of set of structures, we say, okay, our data is good, our structure is well resolved. So this is about the NMR, just one slide. However, the size limit for conventional NMR with these 3D somethings is only 30 kilodaltons. So you cannot investigate the larger proteins using the same approach. You will need something different. And that's the problem with the membrane proteins. So I think you understand that if you have a membrane protein, you need to put it in something that looks like the membrane. And here I show two NMR spectra. This spectrum is for a small protein of 15 kilodalton. It's quite good. You can see it. You don't need to be an NMR expert to understand this is good and this is not. Yes, and if you have 35 kilodalton protein, you get something very ugly. And if you think about the liposomes, which are the most conventional mimetics of cell membrane, its weight is 30 megadalton or even more. So it's 3 orders of magnitude larger than the largest protein which you can study by NMR. For this reason, you need specific small lipid particles which mimic the cell membrane. And here's the picture that you have already seen today. It's from my review of 2017. These are the membrane mimetics that we normally use when we study membrane proteins. It can be detergent micelles, the spherical particles, the bicelles, the discs containing the patch of the lipid bilayer, lipid protein nanodiscs, and some other kind of nanodiscs. So these are the instrument that we have. Okay, it was the introduction. And now the major part. What can we do with membrane proteins, with single-pass membrane proteins, using this approach, using NMR spectroscopy? So the thing that we started with when we entered this area was the structure of dimeric transmembrane domains of the single-pass protein. If we consider the activation mechanism of the single-pass protein, it looks like this. So we have a dimeric structure. It can be dimeric both in the inactive state and in the active state. It is for several proteins, it is so. And when the ligand binds, you obtain the rearranged structure of the extracellular part and the rotation of the transmembrane domains and some kind of arrangement here which launches the activation cascade. So if we think in this paradigm, then we should have two structures, alternative structures of the transmembrane domains, which correspond to the receptor in the active and the inactive state. And it was our first goal in our work, to get these structures and to understand how does it happen. And that's the result of our work for several years. It's a figure which summarizes all the structures that I myself was involved in my career. So we get a lot of dimers of helices. And when we thought about them, we started to group them, to classify. So we classified them into two clusters. The first cluster is at the top. It contains the structures with the N-terminal dimerization and the high crossing angle of helices. And the second cluster is in the bottom. It contains the extended dimerization motif and low angle between the helices. And when we analyze the mutations which can activate the full-length proteins and analyze the structures of the other domains, we concluded that this cluster most likely correspond to the active state of the receptor, and this cluster is likely to correspond to the inactive state of the receptor. And moreover, for two proteins, we managed to get both active and inactive structures of the same protein taking different membrane-like environment. And this provided us with quite interesting idea that lipids may be involved in the signal transduction by the single-pass cell receptors. And this is the way we act further. Yes, when we have the structures of the transmembrane domains and take the structures of the intercellular and extracellular part obtained by the other groups by X-ray or cryo-EM, for instance, we can build the full-length models of these proteins in the active and inactive state and think about the way how they are activated. So that's what we do. Next, the second part. The second part is about the juxtamembrane regions. It's the small helices. So for many proteins, like single-pass proteins, there are not only the transmembrane helices, but also the juxtamembrane, small juxtamembrane elements that can be involved in their protein activity. For instance, for the ARBB receptors, which is very famous protein. For instance, one of the ARBBs, ARBB2, is a very well-known marker of the breast cancer. There are, for these ARBB receptors, there are small cytoplasmic helices, cytoplasmic domains, which are very important because they are, if you mutate them, you lose the activity. If you delete them, you completely lose the activity. So they are needed. And when we made the structure of the protein, which contains not only the transmembrane but juxtamembrane region, and compared it to the structures of the protein without the juxtamembrane regions, we see that they are different. So if we just elongate a little bit our protein and get additional domains, we can get the more native state of the transmembrane part. That's what we were interested in. Another example is the toll-like receptors. And if you analyze the hydrophobicity of the toll-like receptors, you see that apart from the transmembrane domain, you always get the juxtamembrane region, which is also very hydrophobic. And we calculated several structures of toll-like receptors. They are shown on this slide, and for all the objects, we have this juxtamembrane cytoplasmic helix, which is very similar to what we see with the RBB receptors. So that's how it looks like when how it looks like when you put the protein in the membrane. How do they behave? And then we decided to analyze why do this protein need this small helix, and we made several constructs. We deleted this juxtamembrane region. We replaced it with the flexible linker composed of glycines and serines. And we also scrambled randomly the amino acids in the juxtamembrane helix, so we did not alter the overall hydrophobicity, but we removed some sequence context. And I also did not put it here, but our last construct, it was a replacement of the juxtamembrane region from one protein to another protein of the same class. And what we see that all kind of interventions here completely abolished the activity of the protein. So if we do just very small thing to the juxtamembrane helix, you break the protein. And it is also in agreement with the evolution analysis. So if you take the and if you analyze the evolution of the toll-like receptors and study the conservativity of the residues, so here you see -1 is very conservative residues, red ones are conservative, and blue are variable. And you can see that the juxtamembrane region is just here, highlighted in rows, and is almost as conservative as the globular domain, which is responsible for the activation. And the transmembrane domain is completely variable, almost completely variable. So this part of protein is very important, it's very conservative, and it's needed for something, and it's not just not the linker containing the connecting the transmembrane part and intercellular part. It's something, it encodes some kind of specific interactions which we did not find yet. This was part 3. Part 4 is about the structure of intercellular domains. So apart from the transmembrane domains, we can study the intercellular parts of the receptors if they are not very large. Toll-like receptors are very good objects because they're small. They have small intercellular domains, which is only 20 kilodalton. So we took an advantage of this fact and studied one of the proteins. We get the structure that is shown on the slide, and we also, with the help of the lab of Valentin get the crystal structure of the same protein. And what we found out that they appear different, and there was one major difference in the BB loop. It's somewhere here, very important part of the protein, and in one of the structures we see the formation of the disulfide bond, which is not native, and in another structure, we do not see the formation of the disulfide bond. And when we started to think about it, we came to the idea that these two cysteine residues may be needed for something which is not the disulfide bond formation, but was because inside the cells, disulfides are not normally formed. And we came to the idea that it could be the site for the zinc binding, the binding of zinc ions. And we checked this with NMR. It's also the advantage of NMR. You can easily check something like zinc binding. So we titrated our protein with zinc ions. And what we see that when you add zinc, you see the formation of two new states of the protein. So each peak is stands for one state. And we see the upper form and two zinc-bound state of the protein, and they grow when we add more and more zinc ions. And the binding constant is nanomolar, so very specific. We also did some mutagenesis studies, and we find out that if you mutate one of the cysteine residues, you completely break the zinc binding, and this is residue C667. And when you do these mutations in the full-length protein, this mutation of C667 is the only one that breaks the activity of the full-length protein. So this zinc binding is somehow important for the protein activity. And we also did modeling and proposed the structures for the two zinc-bound states that show up in the NMR spectrum. And using our data, like I showed you previously, we can take the structure of the extracellular part obtained by cryo-EM or by X-ray, take our transmembrane domains and take our intercellular parts and compose the model of the full-length receptor, which we can use for some other kind of applications. Apart from the structures, we can study interactions of drugs with transmembrane domains. So on this slide, I provide two examples. The first example is about the Epstein-Barr virus LMP1. It's a very famous protein. And its structure is still not solved for some reason. I don't know why. We also tried it, but it didn't work because we were not able to express the protein. And so what we do, we took one of the transmembrane domains of the protein, and it is known that this domain can contain important residue, D150, which can interact with pentamidine. And what we did by NMR, we investigate the oligomerization of this transmembrane domain, titrated it by pentamidine, and found out that pentamidine interacts here, and it blocks the high-order oligomerization of this transmembrane segment. Another example is about TRKB. It's a receptor of the nerve growth factor. And it was shown that this receptor is able to interact with the antidepressant fluoxetine. And this interaction is functionally relevant. It alters the it really takes place in the neurons of our brain when we eat antidepressant, and it alters the behavior of our neurons. So what we can do with NMR, we can take NMR spectrum at the ligand, follow the position of peaks in the NMR spectra, and find out which residues of our protein are susceptible to the addition of this ligand. And we can map the interface of the point where interaction between the ligand and the transmembrane domain takes place. So we did it. It's shown here with the red area. And surprisingly, or not surprisingly, mutation of one of the residues here abolishes the physiological effect of antidepressant on the TrkB receptor, which was established by other techniques in this paper, which is not our paper, actually. So what is my major thought here is that with NMR, one can study the structures of transmembrane, juxtamembrane, and intercellular domains of single-pass cell receptors and detect the functionally important interactions that are not observed by other approaches, like zinc binding, like very weak interactions with drugs, and et cetera, or dimerization, for instance. However, it was all about the divide and conquer approach. But divide and conquer approach has its obvious drawbacks, which were understood quite long ago. This figure is made to show that if you divide something and then try to assemble back the functional whole, you do not always succeed. In some cases, you get something not functional, or something that functions not properly. And so it's difficult to assemble the functional whole from the separate parts. And you don't know because you don't know the relationships between the states of the separate domains. If we do not think about the Frankenstein, but think about the proteins. And so instead of dividing, we can try to study the larger fragments of the receptor. So we can try to elongate our protein further and further until NMR stops working. And if NMR doesn't stop working, we can get some data. And that was our concept. And we tried to follow it in our recent work when we studied the neurotrophin receptor P75. This is very peculiar protein because it is covalently linked dimer. It has a disulfide bond in the transmembrane domain, which always makes this protein to be in the dimeric state. And for this protein, it was suggested that the so-called snail tongue model describes its activation mechanism. So snail tongue is this device which people normally use to eat snails. If you add here, these parts come apart. So it's the major principle. So the same is for the P75 receptor. If you add the ligand, it brings together the extracellular parts and it brings apart the intercellular parts. They become available for the partners. Partners bind here and you have the signal. That was the concept. But we were, our idea was to cut here and take the full intracellular and transmembrane part of the receptor and study how does it look like. And we did it with NMR. So it's about the gate and structure and mobility all in one, actually. So what we see here, I'm sorry for Russian script here, I forgot to change it. But what we see here, we see the structure at the top, it's a secondary structure. So we see 6 alpha helices corresponding to this globular domain. And we see mobility here in the picosecond time scale and in nanosecond time scale. So what we see is that we have transmembrane domain, which is very, very slowly moving inside the lipid protein nanodiscs. We have the death domain, its globular part, which is moderately mobile, and the linker region, which is extremely mobile and completely unstructured. And moreover, just if you cut away, if you cut here and investigate this domain separately from this part, you get the same mobility value. So there's no difference whether this protein is linked to the transmembrane part or not linked to the transmembrane part. You get the same mobility and the same state of this, of the protein. And this image doesn't change if you make the dimer or covalently linked dimer or whatever. If you change the lipids, you always get, even if you put the protein into liposomes, you still see the death domain, which is tumbling almost freely in solution. And so this means that motions of transmembrane and intercellular part domain are completely uncoupled. And this model is not working at all, because you cannot transfer something from here to here if these states, these parts of proteins are uncoupled. So we started to think about how to put all the data together, and they came to the idea that there should be any helper protein here. Some third protein which brings together and makes these parts more structural, and it also supports the disulfide bond formation here, because it is not formed if you if you just mix together these proteins without adding some additional participant to the equation. So and this is my final outline of my talk, is that structure and dynamic study of two-domain fragments provides the information on the coupling between the domain states, and this information can be really actually used to understand how the activation occurs. And the take-home message that I would like to deliver is that NMR is a feasible approach, and you can investigate the structure of the small transmembrane domains and larger proteins. And if you have any objects or ideas, we are open, so please come and we will discuss. And finally, my acknowledgements. So this is a lot of people worked in this project, and it's a long going project. In our department of our Institute of Bioorganic Chemistry, and in the Moscow Institute of Physics and Technology, and in Valencia, in China, in Switzerland, and our work was funded by Russian Science Foundation and Foundation for Basic Research. And here is our photo. Not very up to date. It was taken 3 years ago, but it shows our quite nice NMR machine. So thank you for your attention, and I'm ready to answer your questions.
Thank you, Konstantin. I think we have time for one question.
Konstantin, thank you for a wonderful talk. I have probably just a general question. So those single-pass receptors that you showed, like PDGFR, I've read that there's some data that they are used by some viruses, like adeno-associated virus, to interact and enter the cell. So could you please comment about the interaction of those single-pass receptors as the targets of the viruses?
Yes, I cannot comment in details. I know that it is a common thing that cell receptors can be targets of viruses. I think you know the SARS-CoV-2 virus and the ACE2 receptor, which is also a single pass protein, as far as I know. So it's a common thing. I do not know many details about the PDGFR and adeno-associated viruses. I'm sorry. It's not my major area.
We still have 1 minute, and therefore I have educational question. So you showed us at the beginning nice spectra and so on, and the question which I got, and I now recall that I have it from Institute. Do you have some kind of resolution analog in NMR?
We have resolution of NMR spectra. It doesn't characterize the structure, it characterizes the spectrum. Is it good or not? For the structure, we have such a merit as convergence. So we calculate 200 models, then take 20 best, which best fit to the experimental data, and look whether they coincide together. And this is the analog of resolution in NMR. The actual resolution is near atomic, so we can measure the distances between two hydrogen atoms with the accuracy of 0.1 angstrom. So
Okay.
that's the real, but it's not the resolution because we can measure one distance, but when we measure a lot of distances, we have an error of about 0.3, 0.5 angstroms. And that is most likely the actual resolution.
Okay. Thank you again.
Thank you.
So we go on with the next talk. I have a small introduction here because the community of structural biologists was shocked and stressed recently because of the appearance and power of AlphaFold. But time is going, and now we know that there are things which AlphaFold can do and things which AlphaFold cannot do. And I'm very glad to introduce Ivan Gushin from MIPT, who will tell us more about this. Thanks, Ivan.
So thank you, Valentin, for introduction. Thank you to the organizers for inviting me. And yes, I'm going to be talking about the protein structure predictions and strengths and weaknesses of AlphaFold. So as you might know, as you have heard several times already today, our goal as structural biologists, we want to know how the cells are organized at the molecular level. And there are many, many different macromolecules inside the cell, which have very different shapes, very different structures, and their function depends on these structures. So, as you have heard, there are three major experimental methods to determine the structure. It is NMR, crystallography, and electron microscopy. And these methods give nice structures, which are very useful, and every year we get more and more experimental structures, and currently we have around 200,000 structures. However, if you go into a sequence database, in the sequence database currently we have more than 200 million sequences, so there is actually a huge gap between the number of proteins which we know to exist and the proteins for which we know the structure. So we need to do something else in addition to the experiments. And of course, this something else is protein structure prediction. And as with any prediction, you need to estimate the quality of your prediction. So for a very long time, people have been conducting the so-called CASP competition, which is a critical assessment of protein structure prediction competition. And the framework is basically like this. So some experimental people determine the structure, they do not publish it, they tell to the organizers that they have the structure, then organizers announce that they have the structures for such and such proteins, then they collect predictions in 1 month or 2 months, they compare the prediction and experimental structures and publish it. And in the first year when it was held in 1994, the result was probably expected. For very easy targets where you have a highly homologous structure, the success was high, but for medium and difficult targets, the rate of success was very low. Here you can see the correctly aligned backbone fraction. As the years gone by, the quality of the prediction was becoming better and better, and then after maybe 20 years of predicting, the quality became moderate, so even for difficult targets, people were correct in maybe 60% of the time. However, this has changed very dramatically in the recent years with introduction of the AlphaFold 2. So here in the upper right corner, you can see the performance of the best approach during the years. So from 2006 to 2016, the quality was more or less the same. Then AlphaFold 1 was a little bit better, but AlphaFold 2 became much better than everything else before it. And also the quality was close to 100%. So it really was quite an accomplishment and a revolution in this field. And also here at the bottom, you can see the plot which shows for every target in that competition, the blue mark is the quality of the AlphaFold structure, and the yellow is the quality of the second best prediction. And you can see that AlphaFold wins by a huge margin usually. There was only one case where AlphaFold was second best method. So AlphaFold was really a great, how to say, very unexpected and very great advance in this field. Soon after the competition, the authors of the algorithm, they published the paper, they published their algorithm, but also, which is very important, they published the code of the program used to make these predictions. And the algorithm is basically like this. You start with a sequence, you go to the sequence database, you generate multiple sequence alignment, and from this you get some frequencies of amino acids at each position, and also information about covariation of different positions. Then this is fed to a neural network, and then to another one, and this neural network generates you the coordinates of the protein. And then it is usually recycled for several times to improve the prediction. And since it became available to everyone, people have started using AlphaFold in their work, and it turned out that AlphaFold is truly impressive. So you can see here some of the headlines saying that it is a revolution or a breakthrough or method of the year 2021. So indeed, it also matches my own experience or experience of my colleagues, that the quality of AlphaFold predictions is much, much better compared to what we expected in the previous years. So what does it predict and how does it do it? So first of all, what is very important is, besides the prediction of the structure of the protein, AlphaFold 2 also gives you a measure of its confidence in this prediction. So for each amino acid, it produces a score which is called PLDDT, which can be very high or just confident or low confidence or very low confidence. And it turns out that although this is a measure produced by AlphaFold, it really truly tells us about how reliable this prediction is. So if you see deep blue, a very confident prediction, then you can be sure that the backbone is packed like this, like there are no, I believe there are no cases where AlphaFold was confident in its prediction and prediction was wrong. Of course, in natural proteins, and you will hear the talk, I believe, after from Vladimir Zabelskii about disordered proteins. So some of the proteins are disordered, and even if the protein is not disordered, sometimes it has a flexible N-terminus or C-terminus or some flexible loops. And for these loops, you cannot predict a single structure. So AlphaFold will color them in a low prediction, in a low confident prediction. But this is not a problem. This is just a reality of the proteins in nature. Here is a nice animation from Sergei Ovchinnikov, which shows how AlphaFold guesses the correct structure during its refinement. So it starts with some approximate structure and approximate map of the contacts between different amino acids. So it is a probability that two of the amino acids are in contact. And then it reiterates and then finds the correct contacts, correct structure, and it also predicts the error, the expected error of the coordinates. The first consequence of introducing AlphaFold was, of course, to try to see how successful it is at predicting the human proteins. And it turned out that AlphaFold is quite nice at predicting the structures of human proteins. So on the right, in gray, you can see the proteins for which the templates of homologous proteins existed before AlphaFold. So it is around 40% of amino acids or of proteins. And then AlphaFold can reliably predict the structure of maybe another 20 or 25% of amino acids in the human genome. So this is quite an accomplishment. The second, maybe unexpected point is that AlphaFold is even more successful for transmembrane proteins compared to the soluble proteins. So on the left, you can see the, how to say, the average, the frequency of the confidence of structure predictions for transmembrane proteins and for soluble ones. And of course, transmembrane protein can have a transmembrane domain and soluble domain. So if you look at the transmembrane domain of the transmembrane proteins, AlphaFold is half of the time it is very confident in the prediction, and for the rest it is also quite confident. So maybe this is expected because actually the transmembrane proteins are usually either beta barrel, which is not very difficult to predict, or alpha helical, and these alpha helices are more or less parallel to each other. So yeah, while for experimenters it is more difficult to work with transmembrane proteins, it turns out that AlphaFold works with them better. So some comparison with experimental methods. People have compared, of course, the quality of the prediction with some structures which were released after the AlphaFold was developed, and the general conclusion, which you can read in more detail in this paper, is that crystal structures are generally more reliable than AlphaFold, and I will show you why it is the case below. But for NMR structures, quite often the AlphaFold is better, although there are some cases, especially for flexible dynamic regions, where the NMR is on the opposite much better compared to the AlphaFold model. What was also unexpected is that AlphaFold was developed to predict the ordered proteins, structured proteins, but it turns out that it can predict the disordered regions in the proteins on par or even better compared to the best predictors of disorder which existed before AlphaFold. And also some of the disordered proteins, they get, how to say, upon binding to their partner, they fold into some structure, and sometimes AlphaFold can capture the structure and predict the structure of disordered proteins when it comes into contact. The next thing is currently not only human proteins have been predicted by AlphaFold, but also basically all of the sequences contained in the UniProt database or in Interpro already have the predictions from AlphaFold, so you don't even need to use the software. If you need to get a rough model, you can just go to UniProt or to Interpro to search your protein, and you can see the picture, you can download the PDB coordinates, and yeah, it is really convenient and really nice for structural biology projects. One important note is that only monomers are predicted, and I will talk about it a little bit more in the following slides, but if you know that your protein is not monomeric, then this prediction can be a little bit off. Soon after AlphaFold was released, so just to give you some understanding, AlphaFold is a program and a code, and you can download it and install on your computer, but it takes some effort and you need a really nice computer, and then it will work for several hours to get the result for a single protein and single structure. Opposite to that, Google, similarly to Google Documents, there is a possibility to use Google servers, so-called Google Colab service, and people have developed a script to use AlphaFold on Google Cloud, and it works pretty well. You can predict a protein around 1,000 amino acids in less than 1 hour in the cloud, so you do not need a good computer, you just need an access to Google. And yeah, it is now has been published, and it is a very nice, how to say, a wrapper around the AlphaFold program, which I'm personally using much more than the original AlphaFold. And here is just a comparison. So for AlphaFold, you need your own PC. The interface is not very friendly. You do not have much control over the algorithm, and also the sequence alignment step is slow, although accurate. And also you need some third-party tools to analyze the structure. Compared to this, ColabFold can work in Google Cloud. It has a rich interface. It has pretty figures. You can control everything it does. So if you do not need to predict 1,000 or 10,000 structures, and if you only need one, then probably it is much easier to use ColabFold. Also, it is updated very frequently, so yeah, it is also more up-to-date compared to AlphaFold. Now to the limitations of, first of all, AlphaFold predicts only the protein structure, so it doesn't predict any ligands, it doesn't predict water molecules, it doesn't predict cofactors. So people started to develop some accessory tools on top of AlphaFold, and the first of them is AlphaFill. It is a database which collected all the known ligands from PDB, and it combines the prediction from AlphaFold with some known active sites from PDB, and it can give you a model of the active site when it is already present in the PDB. But the AlphaFold on itself, it does not predict the position of ligand, and also if you do not have the correct ligand, sometimes the side chains will be in the wrong orientation. The next step was also quite unexpected. When people started experimenting with AlphaFold, it turned out that it can predict the complexes, protein-protein complexes, quite reliably. And it was not the goal of developers. It was not optimized to predict the complexes, but it works quite well for homo oligomers, as shown here, or heteroligomers, and it works. Of course, you need a simple trick. You need to put some polyglycine linker between your 2 proteins or 3 proteins, but in Google Colab, you now have a nice wrapper where you do not need to think about it. Also, it turns out that AlphaFold predicts complex structures much better compared to the state-of-the-art approaches which existed before AlphaFold. After some time, the developers of AlphaFold from DeepMind, they also released a version of the AlphaFold which was trained to predict complexes, and they claimed that it works a little bit better compared to the original AlphaFold on heteromers. However, when it was originally released, it produced the models with huge, problems in the structure. So personally, I still recommend using Google Colab to predict complexes. But you can, maybe at some point they will improve their own version. Also, if there are not so many complexes in nature, so some people went ahead and just tried to predict all possible complexes in eukaryotes. And this paper came from David Baker lab, and they basically did just that. They sorted through all protein families, and they claimed to have found 600 new structures, 100 of new complexes. Of course, it was validated on some known complexes, but also many of them are new and have not been known previously. What is more is some people have also tried predicting stoichiometry with AlphaFold, and it turns out you can get some information from AlphaFold, although not all of this. Also, you can, in many cases, you can predict the structure of peptides bound to proteins. Although, of course, for some very peculiar proteins such as collagen, the prediction is off, but still you can get some information in most cases. So at the moment there are even more advanced algorithms which work for even very complex, AlphaFold still has a limitation of maybe 1,000 or 2,000 amino acids, and some complexes are pretty large. So people have found some workarounds where you try to predict pairwise complexes and then use some algorithm to predict a large complex from pairs, and it works also pretty well even for large multi-protein assemblies. Here is also my own little experiment in trying to predict stoichiometry of so-called ATPase C rings in nature. These are small 2-helix transmembrane proteins, and they can form homo oligomers of 8, 9, 11, or maybe even 17 subunits. And the idea was simple. If I try to predict a dimer, then I can extract the interface from this dimer and multiply my protein to get a complete ring, and then compare it to experiment, of course. And in my own experiment, I get 9 subunits, whereas in nature it is 8. If I tried some different predictions, I still get 8.5 to 9.5. It is not bad, but also not perfect. If I try to model not a dimer but a trimer, I get 2 different interfaces actually. You can see here it in green. So one interface matches the experimental interface, but the second one is different, so AlphaFold is still not perfect. Another big issue, very big issue for proteins, as you have heard previously, is that most of the proteins which are interesting to us, they have many different conformations. And AlphaFold at present, it gives you some 1 or 5 models, and you do not know to which state this model corresponds. It is just a structure, and you have to decide yourself what is it. And you know that human genome contains, for example, around 500 kinase domain containing proteins, and people have tried, for example, to analyze all of the AlphaFold predictions, and there is one particular conformational change which happens in all of the kinases, and the conformations are called accordingly DFG in, DFG inter, and DFG out. And for this particular kinase family, it turns out that most of the kinases are predicted in the DFG in structure and in DFG in conformation, and not in DFG inter or DFG out. So if you need your kinase in DFG out conformation, then probably you need to use some tricks. The same goes for transporters. For example, some of the transporters have many different conformations, called sometimes inward facing or outward facing, and people have analyzed what does AlphaFold predict for these proteins, and they prepared such a plot where each prediction is shown how, for each prediction it is shown how similar it is to the inward facing conformation or outward facing conformation. And if people use the standard prediction, which uses as many as many sequences and multiple sequence alignment as possible, you get pretty homogeneous predictions which all give you one conformation which is more similar to the inward facing structure. However, for example, if you modify the approach and you take only 16 sequences, you get much more diversity in your structure. You will never get the ideal correspondence to the inward-facing or outward-facing conformation, but you can get the spectrum of the structures. And still, if you didn't have the experimental information, then you would have to decide yourself what does it mean, each of these conformation. People have also done a similar analysis for GPCRs. There are around 800 GPCR structures in human genome, and each structure costs maybe $1 or $2 million. So it would be nice to predict them computationally. And here you can see for all the different GPCRs which have an experimental structure in the active and inactive state. So the black is experimental structures and blue are different computational structures. And sometimes you have several intermediate structure predictions from AlphaFold. Sometimes, of course, you have nice cases where you can predict both active and inactive structure. But still, in most cases, you would profit from an experimental structure. We also did a little bit of our own experimentation with AlphaFold, with GPCRs. So GPCRs are known to contain several so-called switches, where the conformation of one amino acid influences the conformation of another amino acid. And AlphaFold appears to capture some variability in the conformations of these switches, but it cannot, choose the right conformers for each of the switches. So for example, if this amino acid looks down, then these two amino acids should be probably in one conformation. If it looks up, the conformation should change, but this is not the case in AlphaFold. So again, the experimental methods are preferable. Also, we've tried using it for docking, and for one of the receptors for which we determined the structure after AlphaFold was released, a number of, compounds which were known to bind, and the affinities were known and the activities were known. And if you use the experimental structure, you can use binding score to predict the activity. However, if you try to use the AlphaFold prediction, the structure from AlphaFold to enrich your set to select the compounds which are more likely to bind to your protein, the success is much less compared to the experimental structure. So for experimental structure, you will find 5 times more active molecules compared to the AlphaFold prediction. 2 last slides is, of course, the question of predicting the structure. It is opposite to the question of designing the protein. So in one case you get a sequence and you want to predict the structure. In another case you want to have a certain structure and you want to find a sequence which will fold into this structure. So people started developing algorithms based on AlphaFold to design proteins, and they are already very promising. And for me, a much more interesting case is so-called protein structure hallucination. So maybe you have seen these videos where a neural net was trained to see dogs everywhere. So it tries to see dogs in the supermarket here, and you can try to do the same with proteins. You feed a random amino acid sequence into your algorithm, and you try to do Monte Carlo search for something which looks like a protein which will fold into a certain structure. And people did several optimizations, and the proteins hallucinated in this way actually do express, and they do fold into predicted structure, and several new folds were obtained. So it is a very promising approach in my view. So to summarize, AlphaFold is very, very promising. However, there are still many opportunities for improving AlphaFold where it currently doesn't give you a nice answer. First of all is stoichiometry and correct complexes. If you need to predict a certain complex, you can spend several days and maybe you will be successful. But if you want several complexes or some difficult complexes, AlphaFold cannot help you at the moment. If you need alternative conformations, or even more, if you need an ensemble, and if you want to know which structures are more common, which are less common, AlphaFold will not help you. So maybe you should use NMR, or maybe you should use molecular dynamics or something else. Also, effects of mutations are very difficult for AlphaFold because it uses this natural information, and every protein will look like its natural counterpart. And you know that sometimes one mutation is enough to change the fold of the protein or to destabilize it completely, and currently AlphaFold does not predict the changes in the protein structure or protein energetics upon mutations. Also, the active sites are currently a problem because AlphaFold doesn't see ligands, and the positions of side chains will be sometimes completely wrong. And if you need to do docking, then you cannot do it with AlphaFold model. Of course, it doesn't predict the cofactors or ligands or post-translational modifications. Currently, it doesn't work for protein design, so if you tell me I want 5 helices surrounding a heme or some other cofactor, AlphaFold will not do this. It also cannot currently help in drug design. It doesn't estimate the energy. It doesn't estimate the binding to a drug. Also, currently DNA and RNA are not supported, so you cannot predict some DNA-RNA complexes. Model of the cell is currently unreachable, still because AlphaFold predicts a static structure and not a dynamic. And who knows what else AlphaFold cannot do. So thank you very much for your attention. If you have any questions, I will be happy to answer.
Thank you, Ivan. Questions?
Thank you very much. I was mostly interested in the membrane proteins part. So when you show and say that membrane proteins are better predicted by AlphaFold than soluble, you rely on the confidence score.
Yes.
Is it really is it the real quality or just the confidence?
So it is a number which is produced by an algorithm. So of course it has some inherent error to it, but from all my experience and from experience of everybody who I've talked to, it corresponds really well to the actual quality or actual accuracy of the prediction. So in our experience, we had several proteins for which we had structure, which we obtained structure after AlphaFold was released, and actually the model matched really well the structure, and the regions which were low quality, they didn't match. So yeah, it is an estimate, but it is a very reliable estimate.
We have an online question.
Yeah,
Sergey,
sure.
you can try to ask.
Ivan?
Yes.
I don't know if you've seen it, but this is Sergey Strepov speaking. Well, I just wanted to say thank you very much. I mean, it was, in my opinion, an impressive overview of something that, is much being talked about currently. I just have 2 short comments because indeed there are many things that could be discussed here. And the first comment is really that we should always realize, and I think you have given many examples of that, so I completely agree with what you said, but we should always realize that at the end of the day, the let's say protein structure's nature is never ever a single conformation. So it's great to see that both experimental methods and performance prediction methods converge at some point, but there will always be quite some variability because it reflects the natural state of things. So that's the first comment. And the second comment about AlphaFold in particular, I had a little experience using it, and I would maybe like to formulate it. Purely as a practical scientist, I would like to formulate a warning that, we, for instance, at some point I've seen people rushing to analyze AlphaFold models which are based for monomers, as you said. You can easily get them from the web, and in our experience, these models are horrendous. I mean, they're completely wrong, and they're wrong for a very simple reason: is that the modeling, when AlphaFold did the modeling, it didn't consider a key element that this is a multimer. And then, as any prediction algorithm, if you are missing, an essential input to the modeling, that the result is basically not good. So that's very, very true for anything which tries to predict, including AlphaFold. So that's, I think, it's a very important thing to remember. Thank you.
Yeah, thank you very much. I mostly agree, I think.
Okay, I think we have time for one more question. There was a question from Alex.
Ivan, thank you for a wonderful overview. I would be curious, so when you run AlphaFold, the initial step is you need to feed in an alignment. Do you have any multiple sequence alignment? And in principle, the depth of this alignment might be different. So you might use like hidden Markov models. You can try to use profile hidden Markov models to create alignments that would compile different distant homologs. So do you have anything to say on whether methods of actually making the alignment and the depth of the alignment really influence the quality?
So if you use AlphaFold out of the box, so to say, I believe it uses HMM search, and it tries to find as many sequences as possible, and you do not have much control on what it uses. But if you use ColabFold, you can choose a lot of different options. And from the general perspective, from how it works, of course, you prefer to have as many sequences as possible, as diverged as possible, because if you take 1,000 sequences which differ in one amino acid, there will be no correlation or covariation between amino acids. So you want to find as diverse as possible. Sometimes it is not possible, actually. It is not possible if you have some rare virus protein, maybe which doesn't have close homologs, or sometimes you have an engineered protein which doesn't have any homologs in nature. And in these cases, the quality of the prediction is usually worse or much worse, but still sometimes it works even for proteins for which no homologs are known. So yeah, I'm not sure if I'm answering your question. If you design, you do not have an alignment, of course. So yeah, sometimes because it is a neural network, sometimes it works even with a single sequence, but not as reliable as with multiple sequences.
Colleagues, it looks like you have a scientific discussion. You can do it during coffee break, and we have to go on. So, in our next talk about CCP4 cloud, so I think that everybody who ever touches crystallographic data knows what is CCP4. And I think this is a big fortune of us that Eugene Chrysanol agreed to give us a talk about CCP cloud. Eugene, are you here? We cannot hear you right now. So there is some technical problem, probably.
No.
Now I can hear you.
Right. So what I do, I unshare now. I stop sharing. Can you hear me now?
Yes, we can hear you and we can see you, but we
Okay,
cannot
great.
see
And
your
I share
presentation.
again. Let me try it. Can you see something on my desktop?
Yes, we can see your desktop. Now we see your presentation.
Right, and you can hear me well.
Yes, good.
Okay, okay, great. Thank you very much. Thank you very much for introduction. Good afternoon, everybody, and many thanks for the invitation to give this talk. It is a real pleasure to speak at this event. And to be clear from the very beginning, I'm not going to teach you crystallography today, but rather present CCP4 Cloud for online crystallographic computation. a comprehensive course in this subject would take way longer than a single lecture. So my goal is to give you just a taste of this new system and to get you interested enough for moving further on your own. For the background, CCP4 Cloud is made of two words. First being CCP4, which stands for the Collaborative Computational Project Number 4, UK, which I'm proud to represent here. CCP4 is not a single group or a firm, but rather a worldwide collaboration of researchers and software developers in macromolecular crystallography, which was founded more than 40 years ago, indeed. The project embraces a number of academic sites and is coordinated today at the Rutherford Appleton Laboratory near Oxford. Our main sites include core group in Research Complex near Diamond Synchrotron in the UK, RefMac and Phaser groups in Cambridge, model building and graphics developments in New York and Newcastle, and a number of associated projects across Europe. We distribute our software to about 25,000 academic and industrial groups across the world, as shown by density of yellow dots in this map. Red dots show location of our regular crystallography schools and workshops, which are held in various locations. 5 to 6 schools a year, typically, at least before the pandemics. CCP4 is probably the largest software suite in the field, comprised of about 500 different software modules. We provide solutions for all computational stages of structure determination, from data processing down to PDB deposition, as shown in this slide. All programs can be operated from command prompt as well, but yet the most ergonomic approach to structure computation today is through graphical interfaces. And here is where the confusion starts, at least with CCP4, because CCP4 has got 3 of them, which is really a bit too many. The first one, CCP4I, was released more than 20 years ago. It is now outmoded and therefore discontinued. It should be used only for legacy purposes, which is basically to look into old structure solution projects which you have in your lab's history or personal history. If you still use it for structure solution today, it means that you missed most advanced methods developed in CCP4 during 5-6 years or even more so. The interface was superseded by CCP4I2 around 6 years ago here, and about 3 years ago we released a cloud version of our interface system. And obviously we did not do it just for fun, but to mitigate technical difficulties with software distribution, to cope with growing volumes with experimental data and increasing demands for computing power, and also to have a system suitable for deployment at large centralized facilities like synchrotrons. In the ideal scenario, the data should not move from the synchrotron and instead get processed where it is collected. And then all stages, data collection, data processing, structure solution, and PDB deposition become just an online experience, which has very positive implications for data security, longevity, access, and transparency. The cloud paradigm is actually quite simple. Your computer is used not as a computer, but as a communicator, while all data and calculations are held by big machines elsewhere in cloud. And in most cases, you will need just a web browser, and the whole job can be done using just a tablet or even a smartphone. These slides present basically all what you should know about CCP4 Cloud technically, and I'm not going to bore with further details. Instead, let's look around CCP4 Cloud. In this business, connection comes first. And it is very important to connect to CCP4 Cloud correctly. CCP4 setup includes 3 icons related to CCP4 Cloud. The one with gear signs starts CCP4 Cloud Configurator, where you can choose your browser, set a server URL, and also manage CCP4 updates. You don't have to do this. Everything comes pre-configured, but you can change it. The launcher with airplane sign starts CCP4 Cloud just on your own machine. In this mode, all data and projects are kept on your device, and all calculations are also done on your device, and no internet connection is necessary in this mode. Basically, it works a desktop interface, and it works out of the box. Good if you are paranoid about your data and want to have different desktop interface. The launcher with Wi-Fi sign binds your computer with the big, so to say, cloud. This launcher is necessary for using CUD and some other programs with the cloud, some other graphical programs with the cloud. You can always connect to the cloud via direct URL, like to an ordinary website, which will let you run everything but CUD and interactive data processing and molecular graphics. And this is the only option, unfortunately, when you're working from mobile devices like tablets and smartphones, where CUD cannot be run in any case. So, right. So let's assume now that we connect to cloud via the Wi-Fi launcher, like here. Sorry, I missed one slide. Once connected to cloud, you are presented with the login page, where you can also register an account and regain access if password is forgotten. Please do not forget to check tips in the bottom of the screen. Sorry, I messed up with my slides a little bit. Right. List of projects is the first thing that you would see after login. Initially, no projects are in the list, and at this point, you can create a new project with add button, or import a previously exported project, join a project shared with you by another user, or load a project from tutorials library. However, the first thing that you should do at first login is go into your account settings, like here, and choose a proper password instead of temporary one given to you at registration. Also, at this point, you're advised to authorize with third-party software providers like Global Phasing, which will unlock using their programs for you in CCP4 Cloud. Next, check other options in your account and pay attention to the set of icons on top of the screen. They link to additional information such as your CPU and disk waters and the actual use levels. After finishing with the formalities, you are set to work, and you can start your first CCP4 Cloud project. For this, push the Add button, and you are presented with the set of 3 basic options corresponding to different project templates. Auto Start projects selected here are suitable for novice users, but also in routine cases where structure solution is not expected to be a challenge. These projects unfold completely automatically, and they can bring you to almost complete structure. You will still need to polish the last bits, such as final refinement and fitting cofactors by yourself with CUD. Standard template, the next one in this list, gives you the same tools as Auto Start template and more on top of those for coping with difficult cases. Standard projects are developed manually, not automatically, and therefore a good understanding of structure solution process is essential here. Hop-on template provides a quick start in cases when you wish to continue projects from other systems like CCP4i, CCP4i2, Phoenix, or Global Phasing in CCP4 Cloud. Hop-on projects let you to import data, model, and phases in one go and just proceed with model building and refinement. Today, we shall consider the simplest type of projects in CCP4 Cloud, the Auto Start, aimed at providing the highest degree of automation that CCP4 can currently offer. Automation is important. It is a key for getting rid of routine and saving human time, but not computer time. But also for mitigating incompetence, which is quite common these days, I should say. It helps with choosing strategy, tactics, resolving ambiguity, and provides inbuilt expertise in the system. These days, you do not need to be an electronic engineer to operate your laptop, and ideally, you wouldn't have to be a crystallographer to solve a structure. We are working hard to that end. And yet there is no ultimate automation in protein crystallography. CCP4 Cloud, though, offers automation on 3 levels. First level corresponds to the set of automated pipelines, which combine many CCP4 programs in them. Each such pipeline uses from several to tens of different programs working in essentially non-linear way, and this is a great help in solving structures. This picture gives you the scale of variety among similar automated tools in CCP4, which can be very confusing for novices, but it also extends the scope of what is possible to achieve with the system. On the second level, CCP4 Cloud offers a suggestive guidance through structure solution projects. When a task is completed, the next step depends not only on the current results, but also on the prehistory of project development and, in ambiguous cases, on personal preferences. Due to the considerable number of possible scenarios, I said it's 500 modules to choose from, such guidance cannot be fully algorithmized. Instead, CCP4 Cloud includes a learning facility which analyzes structure solution projects with time as it goes and suggests follow-up tasks on that basis. The third level of automation may be viewed as a combination of two previous levels, when guidance is applied with, to the automatic development of structure solution projects. We call them workflows. This leaves users with the choice of very few options, basically workflows for molecular replacement with given or unknown structure homologue, and a workflow for experimental phasing. Special workflow also exists for repeat experiments on the same target with varying cofactors or ligands, and one more for structure refinement with automatic ligand fitting. This part of CCP4 Cloud is in a very active development at the moment, and the number of workflows is likely to change in future. Do you still hear me well?
Yes, yes.
Okay, good. So using automated workflows is supposed to be self-obvious, and I'm going to demonstrate that only on a single example of molecular replacement with known structure homologue now. So we begin with creating an AutoStart project. For that, push button, give a short ID and descriptive title for the project. Select AutoStart, which is normally selected by default, but it doesn't have to be, and simply start it. Now we are in a project page, and we can choose a suitable template for the workflow, which is simple molecular replacement with search model, and then give it data from either local system or cloud storage. Reflection data should be given, merged or unmerged. PDB structure should be given as a structural homologue for this pipeline. Sequence should be given as a seq FASTA or PGR file, confirmed it's a protein. And then ligands, I expect a ligand to be in this crystal, so I give a smile string for generating ligand structure and automatic fitting. Data gets uploaded to cloud, and then workflow starts to work completely automatically. In reality, calculations are not as fast as shown here. That's why it was recorded, but you do not have to be connected to cloud all the time. It will work without you watching it, absolutely. You can see the project branching where workflow tries alternative programs or performs multiple runs for adjusting calculation parameters. And very soon in this movie, it will come to the deposition task, which doesn't depose anything, but instead it prepares MMC files for BDB depositions and even acquires the PDB validation report for inspection. So looking at this picture, we can see that our factors indicate that the structure is clearly solved, but not fully refined. How the project may be taken further in this situation, because we will to bring it down to a depostal quality. So consider now basic concepts behind CCP4 Cloud projects. Projects are represented as trees, which indicate data flows, and that helps understanding the structure solution pathway. Essentially, this is log of how the structure was solved, can be read in human language. If two jobs are connected, then it means that data from the upstream job was used on input in the downstream job. Jobs can see data produced by all upstream jobs in the branch, in their branch of the job tree, but not data from other branches. The project is being developed using just 3 basic operations: adding jobs, cloning jobs, and removing jobs, plus a few convenience functions as shown here. All functions are applied to the currently selected job, number 20 in this particular case, and it is possible to select several jobs in different branches and use their output data in a new job. Let's see how this works in practice. Obviously, all examples are greatly simplified for demonstration purposes. So, firstly, inspect the PDB validation report. Double-click on the job, it opens up. Those are files which you would use in deposition if everything is okay, but now we want to check the PDB validation report before deposition to see we are in red on some scores, so we definitely want to rectify something in our structure to correct on this. We should do it with CUD. Append CUD job to the last ref mark job before the deposition. See that R3 is indeed high. I'm sorry, that was a ref mark job. Now, another tool for visual inspection of quality of density in cloud. We can see some greens, some reds, which we want to correct. So, yeah, add a new job, choose CUD from the menu. All data is loaded automatically in most cases, but you should review what's going on in your job, not click like in this demo. Right, everybody knows CUD, I shouldn't introduce it. Just see where the problematic site is and try to correct it here. I'm not particularly good at coding, but I think something may be done here, like this. Right, everything is green now for that particular residue. Now we want to save the result in coordinate file, like here, in default directory with default name, and simply quit CUD. At this point, data travels back to cloud completely automatically from your machine. The job is finished, and now we should give it a final refinement because modification was done, and we want to recalculate maps, and well, that's the essence of refinement. Another ref mark job. R factors are still high, but they went a little bit down by a tiny bit, but of course the modification was tiny, so we cannot expect a big effect. But this is basically how the structure is going to be refined before you can actually deposit it. A quick look at this picture suggests, I'm sorry, I'm sorry. Right, 64 cloud projects, sorry, my notes, I messed up with them. Right, one important thing to notice that if project was started in auto start mode, any part of it can be rerun with modified parameters. For example, many problems and usually arise from the suboptimal quality of reflection data. Suppose we wish to improve data quality in our project, which requires modification of data merging and scaling parameters in job number 2, aimless. You can see that completeness is only 75%, and we can expect that it does not lead to bring our factors down considerably here. So what can be done? We can clone that job. And change parameters, which I know are more suitable for this particular data. Don't ask me how, it is a longer story. Limit number of batches to exclude radiation damage and set up high resolution cutoff. Make sure we keep it on automatic mode, but we can switch it off if you don't like it, and run the job. Now, all the workflow will develop starting from this point with new parameters for aimless and repeat itself. However, the number of jobs and branching can be now different depending on the intermediate results. A quick look suggests that adjusting data processing parameters resulted in noticeably lower R factors there. And therefore, further project development, in this case, should be done in the new branch, starting from job 44, and not from job 20, as we naively did before. In general, bringing structure to ultimate quality usually requires multiple manipulations with Hood and Triffmark and reruns, as I showed here, similar to what was shown. Other programs, such as ligand making with ACE drug, may be required as well in the middle of this process. There are many questions here, but they are beyond the main topic of this lecture. So you're very welcome to check in one of the CCP4 schools where structure completion, refinement, and model building are covered extensively. What to say in the end of this demonstration? CCP4 Cloud projects can be renamed, exported as a single file for backup purposes, imported back from exports, or shared with other users. And this is a very interesting functionality of the cloud. Users can work on shared projects simultaneously in real time. They can start, delete, rename, and open jobs and see each other's actions in very much like a Google Doc environment. For sharing a project, you would need only login names of users with whom you wish to share it, like this. Only project owner can share or unshare a project, and no resharing is possible. Once project is shared, the other user receives email notification and can join the project as shown here. And the joint projects are prefixed with owners' login names in the list of projects for clear identification and keeping authorship, if you wish so, of the development. Right, so this presentation aimed at giving just the initial knowledge of CCP4 cloud operation principles and scope of its possibilities. There are many details and features which we did not cover, and it is expected that they can be discovered and learned in course of working with the system independently. Therefore, I would like to finish this presentation now with some general remarks. CCP4 provides a complete structure solution package, which can be roughly presented in 10 functional categories, as in these slides. CCP4 Cloud delivers on all of them in a set of about 75 coarse-grained tasks, which are built on top of more than 200 individual fine-grained components. Altogether, they give a full structure solution line from A to Z with a generous choice of various options, which is confusing in best CCP4 traditions. And this makes the development of CCP4 Cloud largely finished by now. Yet it is not completed and probably will never be. We still introduce new tasks when necessary, but rather try expanding existing tasks for keeping the system concise and manageable. It is fair to say that CCP4 Cloud's functional scope and possibilities are equivalent to what is given by ordinary desktop GUIs. This development was also a nice opportunity for us to revise our documentation resources and put them on a new foundation. CCP4 Cloud is really well documented, thanks to Maria, who enthusiastically took it forward. And that was a project itself on an epic scale, I should say. Documentation includes a set of tutorials and demo projects for self-education and use in workshops, schools, and courses. And we do this now on a regular basis. Documentation repository is open for contributions from people like yourself, and everybody is invited to join the project. We are a collaboration, and we rely on feedback and assistance of our users. And here, the possibilities are really endless. For example, if you need CCP4 Cloud for your university courses on structure solution, crystallography, structural biology, whatever, you can put up your tutorials and data on cloud and use them right from there. CCP4 Cloud was designed for distribution and installation at custom locations. Beside the main site at CCP4 Harwell near Diamond Synchrotron, there are a few more CCP4 Cloud instances maintained in various places. They are set up to serve researchers from particular labs, institutions, and industry sites. The last slide is really to say that it is easy to make a sales pitch for cloud computing today, but the choice of particular work environment obviously depends on many objective and subjective factors. You are very welcome to try CCP for cloud. Please just give it a try and see if it works for you. I also want to add that project enjoyed enthusiastic contributions from a number of people, and it makes me a real pleasure to express my acknowledgements here. It wouldn't be possible to create this modern framework without a significant collective effort. And the funding from BBSRC UK and CCP4 is greatly appreciated. Here I end my presentation, and I'm happy to take questions.
Thank you, Eugen. Then we have time for one short question.
Okay, Evgeny, thanks a lot for the very nice talk, for the brief introduction. I actually use CCP Cloud for a couple of years and find it very useful. Thanks once again. So maybe you can shed light on some new features you are going to introduce in the nearest future, like, I don't know, automatic manuscript preparation or whatever.
No, that would be far too much. One important feature is, which I did not touch upon in this presentation, is integration with AlphaFold. We have AlphaFold task in cloud, and using AlphaFold predicted models is seamlessly integrated in all automatic pipelines. You simply don't know about this, but it will check for structural homologs in the PDB and in the AlphaFold database from the EBI. And these days we are also working on including meta universe in this equation. You know, the meta released 600 million predicted structures using technologies similar to AlphaFold, but on own hardware basis, and they are very proud of it. Recent news. Another major thing which we are going to release in about 2-3 months from now is CCP4 Cloud Archive. And here the idea is that in crystallography, macromolecular crystallography at least, for 40 years we were having data, then structure solution process, and then interpretation of this data in form of PDB file. And only this interpretation was deposited into the PDB for a very long time, until 2008, where PDB started to ask for original experimental data in terms of merged and later unmerged reflections. This cuts off a significant part of knowledge which is collected in experiment. Data is precious. It is good that it is collected, but also structure solution projects is a precious knowledge. How the data was interpreted, because many important things may come from that point. So we will come up with CCP4 Cloud Archive, where researchers would be able to deposit their structure solution projects against generated archive ID, and then use this archive ID, let's say, in publications or disseminations, or in reference to PDB codes of their depositions. And we hope that if researchers participate in that voluntarily, it will increase quality of data which we operate with in structural biology. Thank you.
Yeah, thanks a lot. Еще вопрос есть в чате у нас успеем. Вопрос в чате еще есть один. Сейчас. Just 1 second, again, there is a question in chat. Just wait a second.
Okay. I can probably see it for a small group. Do you recommend your server at Harwell, sorry, rather than setting your own server? It very much depends on your situation. You're very welcome to use the central CCP4 cloud at Harwell. But if you are concerned about your data, if your data is sensitive and you do not want to transmit it to public servers, then you may be better off creating your own local cloud. Of course, we guarantee that your data is not sold to third parties. We respect confidentiality and so on. And this is represented not by us, but Science and Technology Facilities Council UK. And still, pharma groups, industry are rather setting on their own clouds in-house.
Okay, thank you, Jen. I think we have stopped.
Thank you.
Okay, colleagues, we are starting our last session, and our next speaker is a very well-known person in field of protein folding, Vladimir Uversky from University of South Florida, with his speech protein structure function continuum. Vladimir, are you hear me?
Yes, I can hear you.
Okay, thank you so much for wake up so early. Good morning, probably, yeah?
Good. Yeah, it's not already very early. It's 8:30 in the morning. It's fine.
Okay, okay. Take your time, please.
Yeah, thank you. So thank you very much for inviting me to give you this talk. And I will be talking about protein structure function continuum, and I will introduce the role of intrinsically disordered proteins in this stuff. So first of all, before moving anywhere further, I'm starting with most important slides. This is my acknowledgement slides. And so after this, people can leave the room. It's okay, because everything else is not as important as this next few slides. For a long time, when I was making presentations about my work, I was showing an acknowledgement slide at the end of my presentation, so with a few names. And then I decided that probably instead of just showing names, I need to show faces, because it will be easier to connect names to faces. So, eventually, I collected a pretty large set of people whom I'm collaborating with. And I have probably 20 slides showing these faces. This is my Facebook of collaborators. I definitely don't have any possibility to discuss contribution of each of these people to my work. So, but trust me, without their contribution, this presentation wouldn't be possible, because it's heavily based on stuff which we are doing together. So keep in mind that I have very large set of interactions, and I will be talking about disordered proteins today, which are characterized by high binding promiscuity and a large number of interactions. And from this slide, so you can see from this slide, you can see that I'm highly disordered. So I had this idea that I will be able to collect all of people whom I'm working with, but in 2016 I published a paper in Autophagy, so which had almost 2.5 thousand co-authors. If I still kept an idea that I will be able to collect all of these people, as my collaborators' pictures, in 2019, I got another hit, so with 1,500 co-authors. And last year, I published second paper in Autophagy, so with almost 3,000 co-authors. So now, as you can see, so I don't have any possibility to create a Facebook of all of these people. Altogether, over the years, I collaborated with more than 1,200 and 500 colleagues from a very large number of research organizations in almost any country which is doing research. So this is my interactome. I'm very, very grateful to all of these people who contributed to this work. And so this work wouldn't be possible without their contribution. Two very important messages here. One, if you like what I'm talking about, so keep in mind that this is not me. So this is all of these people whom I was working with and continue to work with. And if you don't like my presentation today, don't blame them. So don't blame my colleagues, blame me. I wasn't able to deliver a message in the right way. Okay, now, this is an outline of my talk. Unfortunately, I have just half an hour, and I can talk about intrinsically disordered proteins probably for a couple of days non-stop. So what I will do today, I will introduce briefly intrinsic disorder. I will show you how intrinsic disorder can be found. I will discuss functional advantages of intrinsic disorder, and then we'll discuss disorder and protein structure function continuum correlation, which is actually really very important. And then we'll conclude with some final statements. So introduction of intrinsic disorder. It's actually pretty simple. If you open any textbook and start reading about proteins, so what you'll see this classical protein structure function paradigm, according to which amino acid sequence has specific information and knows how to fold into specific structure, and this unique specific structure will be responsible for protein function. So we have a very important information flow from sequence through structure to function. So so this stuff was introduced as lock and key idea. So a very long time ago in 1893 by Emil Fischer, and then it was extended to induced feed. So, but still, all of the stuff was rotating around it. So you need to have a specific structure. So for having specific function. And this is absolutely, I'm using a very, very important idea. And personally, I'm considering it as a big bank of protein science, because as soon as this idea was proposed in late over last century, so it's created protein science field, and majority of knowledge which we gained about proteins by the end of last century, so was based on this structure function paradigm. However, people also recognized that so it's not always the case, and you do have some exceptions, and you have some proteins which don't have structure but which have biological functions. And for a long time, these proteins were taken as a very unique and very obscure and not very interesting exceptions, because they were exceptions. But over time, knowledge was accumulating, and it started to be clear that so it's not really an exception. Probably you have a new some interesting class of proteins. Phenomenon of intrinsic disorder was discovered and rediscovered many times, and it's mostly related to kind of our tendency to use special terms as scientific jargon in order to describe what we are talking about. And if you are proposing a new idea, you will be proposing new terms. People will not be able to recognize these terms, so they will be proposing own terms. And as a result, so we have this paleontology in which you have multiple different names given to these non-traditional proteins. So definitely for many people, it will be very difficult to understand this. So we are talking about similar phenomenon. And this is actually an illustration of the statement. So by the end of last century, all these, and probably more than these names, were used in literature to describe disordered proteins. At the end of last century, three groups came up with this interesting idea that what if these proteins with biological function, so but without unique structure, so what if these proteins are not exceptions? What if they represent a new paradigm and some new class of proteins? So it happened to be the case. And what we know now, after biosynthesis, polypeptide chain can fold, and this protein folding pathway was something which was analyzed by protein scientists for more than 100 years. So because this was a correlation between structure and function. Also polypeptide chain can misfold, and protein misfolding and aggregation, so also were known for a very long time, but they were mostly ignored, so because people were looking for structure. And if you cannot have structure, so you have something which is misfolded and non-functional, so it was taken as an artifact. About 30 years ago, it started to be clear that this misfolding is correlated to human diseases, and as a result, so this protein misfolding now is very, very popular subject for analysis. So in addition to protein folding and protein misfolding, so protein can stay disordered. And as it's shown on this slide, so disorder can be found in multiple different levels. Different parts of protein can be disordered to different degree. Entire protein can be disordered to different degree. So what is also important, none of these pathways represent a dead end. So a folded protein can misfold, and we do know for now very, very well that with age, proteins start to aggregate, and we have development of many age-related neurodegenerative diseases. So this will be a transition from folded to misfolded state. But also misfolded protein can be reconstituted and can be returned into normal state with the use of very strong cellular machinery, so responsible for proteostasis. Folded protein can unfold, and so there is actually a pretty large set of proteins, so which have unique structure but which are not functional, and which need to be at least partially unfolded in order to become functional. So there is a transition from folded state to unfolded state. But disordered proteins, so they can fold, they can misfold, and all of the stuff is going in around. So we have a very interesting circle of structure, disorder, and misfolding in proteins. So these proteins are very abundant. So this is illustrated by a couple of next slides. So here I'm just showing numbers, and then I'll show a picture. So what we did here, so we analyzed archaea, bacteria, single-cell eukaryotes, multicellular eukaryotes. So this was a number of organisms which was included in study. So this is a range of proteins. So within these proteomes, this is an average proteome size. So this is amount of disorder in these proteomes. So first column here shows a very, very simple measure. So we have computational tools which can predict per residue disorder propensity in proteins. And when we apply these tools to these proteomes, we can see that the proteomes on average will contain from about 10% to about 45 to 50% of disordered residues. So next column shows a little bit more sophisticated analysis. Here we are looking for how many of all of these proteins in different proteomes, how many of them will contain intrinsically disordered regions, which are defined here as residues, regions containing more than 30 residues. And as you can see, proteomes on average contain from about 10% of proteomes will contain disordered regions up to 50, 60, 70%. So, but we also have computational tools, so which are giving us a very, very crude yes/no answer to disorder status of proteins. So it's so-called binary predictors. So they're telling us, so if protein is expected to be completely structured to disordered, and when we apply these tools, we can see that amount of this completely disordered proteins ranges from about 3% to about 42%. So this bottom line here represent disorder content in human proteome. It's not just me who is disordered, it's all of us. So, and two very important messages from this slide. Intrinsic disorder is really very common and it's present everywhere. And another very important message, so we do have pretty dramatic increase in amount of disorder coming from simple cells like archaea and bacteria to more complex cells, to eukaryotes. And this is better illustrated by next picture. This red crosses, so are viruses, and I'm not talking about them today. So let's focus on this part of slide. So this is dependence of average fraction of disordered residues on proteome size. And so we have bacteria shown by green circles, we have archaea shown by blue circles, we have single-cellular eukaryotes, brown squares, and pink triangles will be multicellular eukaryotes. So, and as you can see, for nature to go from simple cell, so in eukaryotes and archaea, to a more complex cell in prokaryotes and archaea to eukaryotes, it was necessary to increase proteome size or genome size, but it was also necessary to introduce very high levels of intrinsic disorder. So this is really very important. So in order to be more complex, you need to be more disordered. So now we can ask a question: why intrinsically disordered proteins are so common in proteomes? And answer is actually very simple. So nature is not doing anything just out of blue. So if it introduces something, it will be for functionality for some purposes. So so this graph represents a correlation between protein structure, function, and shows that. So if you will exclude intrinsic disorder, don't consider red part of this slide for a moment, and if you'll be focused just on structure. So what we will have our traditional structure function paradigm, which is shown on top of this slide. So we have protein which is able to fold into structure, which will be responsible for specific function. Or we have this lock and key and unused fit. So this will be two major functions, so described by this feature. So, but now if you introduce intrinsic disorder at any stage of protein life, you can dramatically increase possibility of protein to act. So if you have this old order disorder, new order loop, we will be able to create a protein which is acting as conformational switch. So we have very important functions associated with transition from completely structured but not functional state into at least partially disordered state. So this activation is found in these functions are listed here by red. So this will be part of proteome, so which is able to fold, but intrinsic disorder somehow is for functionality in this foldable part of proteome. So, but in addition to this upper part, so we have also second part of proteome in which amino acid sequence is not gaining specific structure. So instead of existing as a unique structure, so these proteins will exist as highly flexible conformational ensembles containing many different and not very similar structures, which are interconverting at very high speed. And we have functions which are associated with this complete lack of structure. But then we also have proteins which will be able to undergo at least partial folding upon interaction with specific partners. And we do have very important functions associated with this feature, so disorder to order transition. So, and altogether, so we have quite a few different very interesting advantages of intrinsic disorder for functionality. And I try to summarize everything on this slide. It's very busy, and I will not go through all of the details here. So, but as you can see, we have at least two different features which are used by proteins for regulation and controlling functions. It will be regulation efficiency and also having some functional advantages of intrinsic disorder. So, so as you can see, we have multiple different mechanisms, so by which intrinsic disorder can be very crucial. So for protein functionality, so it provides faster kinetics interactions. It allows you to form very stable complexes. It allows you to form very transient, very non-stable proteins. So you have a lot of different features, so which really will be very dependent on presence of disorder in a protein molecule. So, so one of very crucial characteristics of disordered proteins, and which provides actually very solid explanation of why these proteins are very common in signaling, so is given by this ability to fold upon interaction. And this is really very crucial for signaling, because signaling interactions are characterized by a very interesting combination of high affinity, low affinity and high specificity. And so in order to have this highly specific but low affinity interactions, so you need to have this scenario which is shown here. So you have a partner which has a binding groove. So we have another partner which is completely unfolded. So on this unfolded protein will interact with partner, will fold into specific structure. And so you do have now a specific complex formed. So this is very specific interaction, or rather specific interaction. So, but because of energy was spent for folding, so this interaction is not very strong. So having disorder and ability of disorder to fold provides you a possibility to be, for protein to be engaged in signaling interactions. So disordered proteins can be involved in other type of interaction, on which they will be kind of touching in this way of binding partners, so without being engaged in any stable contacts. So, but so you can measure association in this complex. You can measure association constant. So, but you cannot see any specific structural changes in disordered proteins. As it illustrated here by this polyelectrostatic model, so you have a protein so with an active site, so which is able to recognize phosphorylated serines and threonines in binding partner. So binding partner has multiple phosphorylated serines and threonines, each of which has exactly same or very similar affinity for binding site. And as a result, so when we are observing complex, we are seeing such kind of interaction, so kind of plain staccato. And what is important, so no structure is formed. And another important feature here is that so strength of interaction is proportional to degree of phosphorylation in a binding partner. And a higher degree of phosphorylation, stronger interaction. And if you have a particular threshold, so in this particular case, it's 5 phosphorylated residues. If it will be below 5, complex will not form. If it's above 5, it can form, and it will be stronger if you have more phosphorylation sites. So now, if you put the stuff together, so we have 3 different modes for protein interactions. So we have, based on disorder, so we have induced folding, in which disordered protein will fold upon interaction. So we have proteins plain staccato. Once again, I'm showing the stuff here. But also we have 2 clouds interacting with each other. It will be this type of interaction. Intrinsic disorder also has clues for answering very crucial questions which cannot be answered using structure. And one of this will be alternative splicing. Alternative splicing is a very common mechanism in eukaryotes, which allows us to increase our proteome size, keeping genome small. And alternative splicing is based on this so-called mosaic structure of many eukaryotic genes, in which you have coding regions and you have non-coding regions. And during normal splicing, all non-coding regions are removed. So we have our DNA, so we have mRNA, so which will be produced from this DNA, and all non-coding regions are removed. And this will be canonical form, so which will be translated in a polypeptide sequence, which will be able to fold into a structure. So when we have alternative splicing, in addition to non-coding regions, some coding regions also will be eliminated. So and as a result, we have a new isoform for protein, so which is not keeping this red piece. And question now will be, so will this protein be able to fold? And I was working on protein folding before moving into disorder for a very long time, and very, very simple answer here. So in vast majority of cases, when we have sequence which is not containing piece, so significant piece of sequence, so it will not be able to fold correctly if it was foldable protein. So now it's a really very interesting conundrum. So we have probably about 80% of genes in human, so which undergo alternative splicing, and we do know that kind of most probable output for protein to so missing some pieces of sequence will be protein misfolding and associated diseases and all of these are other troubles. So question is how this stuff works. And answer is really very simple. What is alternatively spliced is mostly coding for intrinsic disorder. So you can have no structure, you can take any piece of no structure, remaining no structure, so you will not be kind of feeling the stuff, you won't be engaged in so folding anyway, so and you don't have this conformational catastrophe. So, but it also gives you a possibility to provide a very strong and interesting control of protein functionality. So, and I'm illustrating here the stuff by protein BRCA1, breast cancer protein 1. This is a large protein, about 2,000 residues. Structure is known for first 100 residues and for last 200 residues. And the middle part, so which is about 1,500 residues, so it's completely disordered. So protein is engaged in multiple interactions. Protein has multiple phosphorylation sites. So as you can see, a lot of interactions will be located within disordered regions. For all phosphorylation sites are located inside of disordered region. So now I'm showing alternatively spliced isoforms so for this protein. So this is canonical form, and each dashed line will represent regions so which are taken out by alternative splicing. And so this kind of shaded area will show you how functionality of this protein will change if alternative splicing is engaged. So, so we can dramatically change what protein can do if you add alternative splicing, so combine alternative splicing with intrinsic disorder. And this is actually was really very crucial combination so for creating multicellular organisms. So another conundrum will be post-translational modifications. We do have about 520 kinases. So we have about 250 phosphatases. So this will be enzymes which are adding and removing phosphates from proteins. So about 75% of eukaryotic proteins are phosphorylated. Many of these proteins are phosphorylated at multiple sites. So if you put all of this stuff information together, even keeping in mind that kind of assuming that so you have just one phosphorylation site per protein, so what you will see that so for each human kinase, so you have 30 different substrates, and for each human phosphatase you have about 60 different clients. So which means that so you have for each lock you have multiple keys in both cases. And so that actually tells you that so probably lock and key is not applicable in this particular case. And so in reality what is happening, so we have this very crucial role of intrinsic disorder sites which are modified it's not only phosphorylation, it's probably any type of enzymatically catalyzed modification of protein. So these sites of modifications, so they will be located inside of intrinsically disordered regions, so which will allow a region to adjust into active site of kinase. So definitely phosphorylation and all other PTMs, post-social modifications, so will not be possible if you don't have intrinsic disorder. So it's also applicable to protein-protein interaction networks in which you have protein hubs which are engaged in multiple interactions. And so some hubs can have a few thousand of interactions, and definitely for this stuff to work, intrinsic disorder is crucial, and intrinsic disorder will be present on either in hubs or in hubs and partners or in partners. So you need to have disorder at least on one side of this equation. I'll give you just one very interesting illustration here about how disorder can be used. So for protein functionality, this is P53. Very well studied protein. It has a lot of information. In PDB, it has multiple PDB entries. So, but there is no any structure for full-length protein because it's actually pretty disordered. It has about 50 to 60% of residues which are disordered. So what you will find in PDB, you will find small pieces of P53 co-crystallized with Vladimir partners. So pieces of P53 are shown by colored wiggles and partners are shown by this cyan clouds. And I'll spend a little bit more time on this last part of protein C terminus. Same region is able to interact with 4 different partners and will fold differently upon interaction, so to different partners. So which means that so with one region which is content dependent, which can fold in content or partner dependent manner, so you can control multiple partners. And so this is actually really very crucial because P53 is a very major regulator of a lot of activities in proteins. So now let's move into protein structure function continuum. So because time is running out. So first of all, these proteins are characterized by very strong structural heterogeneity. When we just started working on disordered proteins, we were looking for these noodles, highly disordered proteins without any structure, so because you were trying to find examples of biological activity, so without any structure, to oppose biological activity with a well-folded structure. So almost instantaneously when we start to look on this stuff, it started to be clear that this blue-red or black and white distribution of proteins is not quite right. So because in addition, instead of being completely structured and completely disordered, so we do have this continuous spectrum of protein structures and conformations. On one end, definitely you have something which is very structured. On another end, you have something which is completely disordered, but also you do have different parts of protein or different entire protein which can be disordered to different degrees. So you do have this very remarkable structural heterogeneity. So within one protein you can find fold-ons. So these are not domains. So these are smaller units. This is independently foldable units in the protein. You can have inducible fold-ons. So this will be regions which are disordered but which can fold upon interaction with specific partners, as illustrated by P53. You can have inducible morphine fold-ons. This will be regions which are able to fold differently upon interaction with different partners, and it will be C-terminal part of P53. You can have non-fold-ons, regions which are not foldable but functional, semi-folded, semi-fold-ons, and unfold-ons, so regions which are structured but not functional and need to undergo at least partial unfolding in order to become functional. So, all of the stuff can be found within one protein molecule. Each of these differently structured or differently disordered parts within one protein, so they could be responsible for different functionality. So which means that in one protein, instead of one protein, one structure, now we have a very, very complex correlation because of addition of intrinsic disorder. So I'll skip this slide, I will go over there. Instead of traditional correlation, so one gene, one polypeptide chain, unique structure function. So because of intrinsic disorder combined with alternative splicing, post-translational modifications and everything else, so we have a possibility to produce from one gene multiple different proteoforms. The proteoform will be a form of protein produced from same gene. And so you can have multiple variabilities here. So you can have basic proteoforms, so which are related to presence of intrinsic disorder and flexibility in proteins. So you can have inducible proteoforms, which will be result of modification of protein by alternative splicing and post-translational modification of some mutations. And also you can have functioning proteoforms, so which will be a reflection of protein functionality, induced changes in proteins. And all of the stuff can dramatically change how protein looks and how protein acts. And as a result, so you have this structure function
Vladimir,
continuum.
excuse me, we're out of time.
So it's my last slide.
Okay.
The structure function continuum, in which from one gene, so you can generate multiple different forms because of post-translational modifications, mutation, alternative splicing, and combination of all of these factors. So, and so definitely will tell you, so from one gene, so because of all of this stuff, you can have very large variability in structure and function. So, so this is a new concept in protein science, and we have explanation for many queries and many mysteries which were not explainable before. So, and I'm done. So intrinsic disorder is very common, so it's predictable, it's related to structure function continuum. So, and so we should kind of take this phenomenon very seriously while working with proteins. Thank you.
Okay, thanks, Vladimir. Let's thank our speaker for the overwhelming lecture. Colleagues, we're out of time. Maybe one short question, if there is. Okay, so I have one. Vladimir, please, can you comment? We listened to the very nice lecture concerning the AlphaFold possibilities. Does it somehow affect your research, your interest?
So AlphaFold definitely creates you a very nice view of protein. So very big advantage of AlphaFold in comparison with everything else. It gives you a picture of structure and disorder. And disorder is shown in AlphaFold as these yellow, orange noodles. So, from viewpoint of looking at this stuff, it's really very nice. So accuracy of AlphaFold prediction of intrinsic disorder is not the best. We have much more accurate computational tools for doing this stuff, but big plus of AlphaFold, so you can see it. But there are multiple minuses in relation to intrinsic disorder. So AlphaFold gives you an impression that so this is a static picture. So, but in reality, so when you will look at proteins with intrinsic disorder, all of these yellow and orange noodles, which are shown as a string in AlphaFold, so they will be actually it will be conformational ensemble, so which is not shown by AlphaFold. So and also AlphaFold is sensitive to any type of structure, which means that so it will be also able to find some disordered regions which are able to fold upon interaction with partners, and quite often AlphaFold will predict this region as structured. So, but in reality they are not. So they are structured only upon interaction. So it's absolutely outstanding development. It's really very cool. So, but it's need to be better connected to intrinsic disorder.
Okay, thanks once again. So we are going to our next speaker. I would like to introduce Professor Alexey Shaitan from Moscow State University, who is a specialist in integrative structural biology. And one short note, I noticed that, Alexey, you have a birthday today, isn't it?
Yes, that's correct.
Okay, we have to celebrate him, I guess, yeah?
Thank you, Costa.
Nice present for all of us. Okay.
Yeah, so it's really an honor to spend my birthday in such a wonderful audience. And I would like to thank the organizers for inviting me, and the program is really wonderful. There have been a lot of interesting talks. So my talk would deal with application of structural biology to understanding genome functioning, understanding different protein DNA interactions. So let me give a few slides of an introduction. So our genomes, as you know, are contained in the structure called the nucleus of eukaryotic cell. And this structure is, the nucleus is probably one of the enigmatic structures that we still have to understand. So the size is around 10 micrometers, and for instance, for the human genome, around of 10 meters of linear DNA are all compacted in this small space, and they are functioning. So the genome is functioning in this small nucleus. I think that there are like three probably main challenges in understanding the structure and dynamics of chromatin in the nucleus. So the first challenge is that the spatial range of the nucleus, around 10 micrometers, it falls below the range that is accessible to optical microscopy. And from the other side, structural biology methods like X-ray, crystallography, and so on, they deal with structures on the size of nanometers, tens of nanometers, probably hundreds of nanometers. And that's one of the challenges in understanding how the structure of the nucleus is organized. The other challenge is actually the dynamics. So we have heard a wonderful talk by Professor Uversky about the disorder. There's a lot of the disorder and order at the same time in the nucleus. And the third challenge is that even small changes in the, let's say, amino acid sequence of chromatin proteins, they may manifest in quite some big changes in the overall structure and functioning of the nucleus. So what we currently know about how the nucleus is organized. So the pioneering work of Don and Ada Ollins was around 50 years ago when they first saw those tiny beads connected by the DNA when you extract the contents or the contents of the nucleus and do electron microscopy. So they call this structures the new bodies, and now we know that they are called nucleosomes. So then this beads on the string model is compacted in the different loops, different domains, and then they are compacted into chromosome territories. And we do not have complete understanding currently of how this whole thing functions. Although a lot of progress have been made through the combination of different methods, starting from X-ray structures, cryo-EM, NMR, different biophysical methods, including genomics, microscopy, and so on. Moreover, to understand how the DNA is compacted inside the nucleus, we have to use a whole range of concepts. Not only understand how the 3D structures are organized, we need to take into account the conformational dynamics, the disorder, the topology of the DNA, different soft and active matter concepts like phase separation, where there are soft physical forces, they are acting to attract different chromatin domains. So in the next part of my talk, I will focus on this part of the of this scheme. We'll talk about the nucleosome and supranucleosome organization of chromatin and different methods that can be used to understand the conformational dynamics and structure of chromatin at this level. So let me start with showing you how the nucleosome structure is actually organized. So actually, there are 4 histone dimers, and if you print those histone dimers on a 3D printer, you see that they nicely fit into this compact structure. And the molecular representation of the structure can be seen here. This year, we celebrate a 25-year anniversary of the day when the atomic resolution structure of the nucleosome core particle was solved with X-ray crystallography. And because the structure is rather big and dynamic, it's around 200 kilodaltons, it took almost 10 years to crystallize the nucleosome and to be able to resolve the structure. So you see the DNA is wrapped in a left-handed superhelix, around 150 base pairs. And here we see the positively charged histone core composed of this histone dimers, H3, H4, 2 of them, and H2A, H2B,
two of them. So there are 14 binding sites everywhere where the minor groove of the DNA touches the histone octamer surface. There's an arginine side chain that basically serves as a cog on a sprocket that holds the DNA in place. So for some time, due to this crystal structure, people were thinking about the nucleosomes as more or less compact and rigid bodies that were used to compact the genome. However, we understand that in chromatin a lot of the dynamics happen, and nucleosomes should, to some extent, somehow contribute to this dynamics and deal with this dynamics. For instance, the transcription when the polymerase approaches nucleosome should somehow either overcome nucleosome or just displace it. Some other examples, right? Transcription factors. If a transcription factor wants to bind to the DNA, the nucleosome impedes these processes. So for instance, the nucleosome, this is a concept from John Whedon from late 90s, the DNA has to unwrap from the nucleosome in order for the transcription factor binding to happen. There's also a lot of ATP-dependent machinery that tries to remodel nucleosomes, to move them along the DNA, and so on. So here is a slide with some overview of different dynamical modes that have been proposed for the nucleosomes. So here you see the compact state, right? In the compact state, there's some flexibility of the linker DNA. There may be some motions of the histone octamer along the DNA. There might be DNA unwrapping, the loss of some histone dimers, and so on. What became apparent quite a few years ago and was to some extent really interesting is that different small modes, small dynamical modes that are embedded, so to say, in the nucleosome structure are important for its functioning. For instance, just a few years ago, with cryo-EM microscopy, it was shown that the remodeling complexes that can move the DNA along the histone octamer, they use some small distortions. So they induce small distortions of the DNA that actually propagate around the histone core. Now it was shown that introducing some rigid links like disulfide cross-links inside the histone dimers somewhat surprisingly would impede these processes. This means that even the intrinsic dynamics of this histone core is important for many functional processes. So how can we study the intrinsic dynamics of the nucleosome? So with X-ray, we usually get a static structure. With cryo-EM, we get a static structure. However, we can get some complexes of nucleosomes with other proteins, and in this way probe the response of the nucleosome to binding with different proteins. There are, of course, a lot of biophysical methods like small angle scattering, footprinting, but they do not give us an atomistic picture. Now, with respect to these histone tails that emanate from the nucleosome, we usually do not see them in X-ray or cryo-EM. However, they are important both for different epigenetic modifications and for interaction with different chromatin proteins. And here, NMR methods, they are very helpful. So all in all, combined with molecular modeling, molecular dynamic simulations, this is the arsenal of what we actually can use. However, in the next part of the lecture, I would like to present you a new interesting approach to studying to understanding the conformational dynamics of the nucleosome, which we call UNMAS, or automated analysis of PDB structures. So you know that due to the cryo-EM revolution, the number of big structures in the PDB is growing, and nucleosome is a relatively large structure. So here you see the timeline of different structures appearing in the PDB. So we start in 1997 with only one structure with resolved with X-ray crystallography. And then starting from 2017, more and more structures resolved with cryo-EM are appearing. And now, all in all, we have around more than 400 nucleosome structures in the PDB. So you see here that the median resolution, of course, is falling because the quality of the cryo-EM with the big structures is not that good as it was with X-ray crystallography of compact nucleosomes. But on the other hand, we get more and more complexes of nucleosomes with other proteins. So we thought, how can we make use of this big data, of this amount of information? And we decided to develop some automated methods to analyze those structures. So this comes with the database that we have developed. It's called the nucleosome database, which basically presents the results of the automatic analysis of our pipelines. It classifies all the nucleosome structures. It identifies the sequence. It superimposes them. It orients them in a correct way, and so on. So let me now tell you about the approach that we use to implement this analysis of the nucleosomes. So structural biologists, they are very familiar with superposition of the structures using the root mean square deviation, like RMSD. So if you have 400 structures and you superimpose all of them, you do not get much way somehow to analyze the resulting information. So what we decided to do is to identify some key axes of the nucleosome. So it turns out that the nucleosome has two principal axes. The one is the so-called the pseudosymmetry axis. Because the histone core is composed of two H3H4 dimers, two H2B H2B dimers, it intrinsically has this twofold symmetry axis. If you rotate the nucleosome around this axis, you get you basically superimpose the nucleosome onto itself. And the other axis that can be introduced is the superhelical axis. So if you present this DNA as a superhelix and solve the mathematical equation, feed the mathematical equation for the superhelix, you can get this axis, the superhelical axis. And once you have defined these two axes, you can automatically define the third axis, and you get what we call the nucleosome reference frame. And in this nucleosome reference frame, you can already analyze, for instance, the 2D projections of the C-alpha atoms of the key structural elements of the nucleosome. So for instance, here, the long alpha helices of the histones are depicted with respective colors. And then this is the DNA. Moreover, because the DNA, you can analyze the position of the DNA with respect to this cylinder. You can model the histone octamer with the cylinder. And then you can basically calculate the rotation angle of the DNA with respect to surface of the cylinder and get this nice profiles of the conformation of the DNA along basically the nucleosomal DNA. And using this approach, we can start comparing structures. So first of all, for instance, here you see the several, from several structures, the conformations of the DNA in this 1D plot are superimposed. And you're starting to see that there are some deviations in the conformations of the DNA in these parts, meaning there is some conformational dynamics. I would also say that this approach allowed us to identify different errors in PDB. So once you start analyzing all the structures in the PDB, sometimes we would see that, for instance, the DNA sequence would just be placed incorrectly. Now, with respect to the analysis of the conformations of the histones, you can superimpose nucleosome structures in the reference frame from different studies, select structures with the appropriate resolution, and then try to identify some parts of the histones that are more dynamic. So in this way, we had some idea that some ends of one of the alpha helix demonstrated more dynamics, and we thought that it may be connected somehow with the conformational heterogeneity of DNA conformations within the nucleosome. So now I will switch gears and show you how a complementary approach, basically molecular dynamics simulations, led us to identifying what kind of conformational motions were hinted by the analysis of the PDB structures, and how basically the functional motions of DNA along the nucleosome could be further analyzed. So with all atom MD simulations, you basically use classical mechanics and equations of motions, the Newton's second law, to study the dynamics of those systems and all atom representation. So here you can see a nucleosome in a box of water. You should add ions because this is a charged system, so you would use 150 millimolar salt of sodium and chloride. And using our supercomputer at Moscow State University, we for the first time were able to obtain record-long trajectories of molecular dynamics. They were 15 microseconds long. So before us, the trajectories were usually on the order of microseconds. And it turned out that getting to this longer scale, almost one order of magnitude longer, actually allowed us to see some new conformational transitions. So first of all, let me show you this. I would not show you the actual videos, but here is just the superposition of different frames in MD simulations. So here you see the nucleosome and the histone tails, and this kind of superposition of dynamic, disordered histone tails. Actually, the lady, Dr. Caroline Luger, who solved first the X-ray structure of the nucleosome, when she saw this picture, she tweeted in Twitter that this nucleosome spaghetti. So this is just the same as Professor Versky was showing you spaghetti for the disordered proteins just some moments ago. So yes, it's really some spaghetti here, but then you have this part that is pretty more globular. Now, if you remove the histone tails, we could see actually the process of DNA unwrapping and rewrapping. Now, with this analysis of MD simulations, we could actually understand how this unwrapping, rewrapping process happened, the kinetics of this process, and more importantly, what are the key interactions, what are the key interacting residues of the histones that serve as anchors that hold the DNA tight around the nucleosome, and what contacts should be ruptured for the DNA to be unwrapped. So let me probably skip this slide. So the next thing that we wanted to analyze is to see, okay, so we saw some conformational dynamics when we analyzed all the PDB structures. So does this somehow compare with what we would see in MD simulations? So it turned out that in MD simulations, if you superimpose the projections of the C alpha atoms of the key helices, you would indeed see some quite substantial conformational dynamics. And if you, for instance, superimpose two most different conformations, the length, the displacement of some of the ends of the alpha helices would be as high as around 7 angstroms. And this is actually this part of the alpha helix that was kind of hinted during the analysis of the PDB structures, that it had some, let's say, enhanced conformational flexibility. So what we could see next, actually, is that during the MD simulations, we could actually see how the DNA conformation would switch from one conformation where the DNA would be compressed by one base pair to another conformation where the DNA would kind of relax. So this plot nicely shows you the difference between the two conformations. So why those DNA conformations are important? They are important because apparently the sliding of the nucleosome along the DNA, which is actually important for the positioning of the nucleosomes and what the chromatin machinery does during different genomic processes, it happens in this kind of what we called a corkscrew mechanism combined with this inchworm mechanism. So in inchworm mechanism, something should, compress locally and then extend locally, and in this way it would propagate. But because the DNA is like a screw, right, like a corkscrew, it has simultaneously to rotate around the histone octamer. Now, what we actually observed in MD is how this process of DNA translocation along the histone octamer actually happens, and how it is coupled to the bending of certain alpha helices in the histone dimers. So it turned out that actually the bending of this alpha helix that I've shown you previously is important for the DNA phosphate to move through this binding site. So here you see a DNA nucleotide, and the helix is in its normal position. Then this alpha helix bends towards the center of the DNA, and it allows some lead space for this nucleotide basically to slide along the DNA further towards the center of the nucleosome. So further, we try to investigate whether this kind of dynamics is functionally important in other cases. So it turned out that when you analyze MD simulations, you can do some principal component analysis, identify those motions, and then try to use these collective variables to analyze how the conformation of the dimer changes in different situations. So first of all, it turned out that the DNA interaction with the histone octamer, it causes some conformational rearrangement within the histone dimer. And it turned out that this conformation, so here it's depicted as some angle between the alpha helices. First of all, it changes once the DNA translocates through the nucleosome. And second, in different variants of histones, so we know that there's not only a single histone set in human genome, there are also some variant genes that are important for marking the genome locations through the genome, where some nucleosomes, for instance, has canonical histones, other has variant histones like H2Z. So the changes in the amino acid sequence, it also changes the conformation and the conformational dynamics of the histones. So I think my time is running to the end. I hope I have shown you how we can combine molecular dynamics and mass analysis of PDB structures to gain some insights into the details of nucleosome dynamics and try to link those details to nucleosome functioning. I would probably add that this dynamics is also important not only at the level of just one nucleosome, but with coarse-grain modeling we can extrapolate those changes in nucleosome conformations to try to model further supranucleosomal structure of chromatin fibers. And it turns out that the nucleosome dynamics are important basically for understanding the flexibility and the conformation of nucleosome fibers in the genome. So I would like to thank my colleagues and PhD students working with me on this project. This is Grigory Armiev, Anastasia Knyazeva, and Anna Gribkova. Without him, those studies would not have been possible. And I thank you for your attention.
Okay, thanks, Alexei. Thanks for being in time, first of all. Okay, so we have time for 1 or 2 questions, if any. Okay, well, maybe I have one. Probably I missed at the very beginning. So there's a very nice research. You have used a lot of different methods. What is the major scientific goal of this study?
So the major scientific goal is actually to understand how the details of nucleosome dynamics, right, they are related to nucleosome function. So for instance, we show that some modes of the nucleosome dynamics, they are really important for the movement of the nucleosomes along the genome. And then this can be extrapolated to understanding how different epigenetic modifications changing in the protein sequence actually alter the movement of the nucleosomes and genome functioning.
Okay, thanks a lot. Any questions, colleagues? Okay, let's thanks once again.
Thank you.
Alexei. And we're going to our next speaker from Germany, Dr. Gleb Bourenkov, who is one of the, I guess, key person on the beam line in the synchrotron in Hamburg. Gleb, do you hear me?
Yes, yes, I hear you.
Okay, please take your time.
Can you hear me? So you.
Yeah, for sure.
Okay, thank you very much. Thank you, Konstantin and Valentin, for inviting me to this presentation where I decided to kind of show some of the things we are doing at the synchrotron source, probably beyond the usual, or somewhat beyond the usual portfolio of high throughput macromolecular crystallography. So the source that basically we are using here is the Petra 3 operating somewhat longer than this. There's an echo somewhere. I don't know if I can do anything to cancel it. Okay, right. So basically, it's a former high energy physics machine rededicated to synchrotron source. It's still a third generation synchrotron source. Probably it is the last of the third generation. A synchrotron source which still provided gain in substantial gain in brilliance, or reduced emittance by a factor of 4 to 5 relative to the previous high energy synchrotron rings. And well, basically as of today in 2028, this probably will change to another figure, to a higher figure at some point. So it's planned to be restarted or rebuilt and restarted as a 4th generation low emittance synchrotron source. But what we're working now with is a small sector of biological structural biology beamlines, and basically in this part of the Max von Laue experimental hall occupied by EMBL, we're using 1.5 straight section of Petra 3 to operate one small angle scattering beamline, very famous, associated with Dmitrii Zabelskii until recently. And then 2 crystallography stations sharing one contact undulator sector P13 and P14, and this now exists an extension of P14-2 or T-Rex. Beamline is basically right next to this beamlines. We're operating so-called integrated sample preparation and characterization facilities, where for crystallographers, for instance, like basically the complete cycle from purified protein through crystallization, through robotic crystal harvesting and mounting, and up to delivery to the beamline is offered to user to be performed. And as I already kind of tried to say, most of the day, this MX beamline P13, P14 operate in high throughput mode as hundreds of other beamlines throughout the world. So they are rather performant. We can basically process or basically collect the data set of a normal cryogenically mounted crystal in a cycle of less than 2 minutes, 100 seconds of which goes into crystal handling and data collection. In 20 seconds, I almost not touch this part of our beamline operation anymore in this talk, and I concentrate on the P14, which is which is somewhat more elaborate and performs more kind of interesting and I can say future-oriented functions. So basically, the build of P14 is, well, basically, well, you know that a typical crystallography beamline in the world would consist of the source, some focusing elements like focusing mirror basically and the end station. We are collimating the P14 white beam using the compound refractive lens transfocator, so that we can use this collimated beam on a sample. We can use optionally the Kirkpatrick base system for microfocusing, for achieving really tiny micrometer sized focus on our sample optionally, and the switch between these two modes, the collimated mode and the microfocus mode, is performed in a minute and accessible to any user on any experiment. And then we also basically refocusing the beam to deliver it on the second interaction point in a second T-Rex hatch. So what we are doing with those beams, the first, well, basically application is a collimated top hat beam, as we call it. So it's a precisely defined beam tailored to the crystal, which are in high demand for high precision crystallography, and in particular of also on high precision crystallography of large complexes that we were able to start with these conditions that I will show later. The microbeam application, well, related actually closely to the top hat beam application, is actually not the application in not in structural biology, but in X-ray imaging, that we found out that actually using our precisely defined and basically very well defined X-ray beams, our like precision goniometry that we are kind of used to in crystallography, and all our automatic sample handling kit, we can do quite efficiently experiment in X-ray tomography of microorganisms or tissues. Or this is again, like I said, well, it's a topic which starts occupying quite a portion of our beam time and activities around the beamline, but I won't touch it today anymore. And basically, we have our microbeam option is most of application of it, as it turns out, now going into time-resolved crystallography. So, well, it's started basically. So the serial synchrotron crystallography started at Petra a little bit earlier, literally like a few months earlier than on many other on all other synchrotron, almost all other synchrotron sources in the world. So basically, in early 2010s, we exercised various methods, various sample delivery schemas. So this is the cryo method in particular, here shown in the middle, basically aiming into delivering into the beam microcrystals in a cryo-cooled crystal suspension via serialized helical scan, and thus collecting like static crystallography data. This is not time-resolved crystallography method. Went through like a very long history of development and basically making it accessible and automated to the extent actually similar to what we have for normal standard single crystal crystallography. Basically just here's the beam conditions also as of now, so you see that we can kind of that we need to expose 25 millisecond per crystal in this beam in these conditions at cryo temperatures and half a millisecond at room temperature. Here's the one basically the most probably prominent application example of this method. So this method is like was invented for the case of like unoptimizable crystal targets, where basically only microcrystals and only very weakly diffraction crystals can be obtained, and in order to obtain any diffraction, any interpretable diffraction out of this crystal, very high dose exposures are necessarily required. It turns out in practical life that basically when you need 100 crystals of that on a crystal mount, and when the preparations are basically deliver such a such amounts of crystals, it's probably not far away from, just like one step away from optimizing and getting crystals to much to the range much more accurate and single crystal data can be obtained. But in this particular case, it's this target really resisted optimization in crystallization or in cryo-EM preparation or in any other structural biology methods for years. It is important. It is an important target. It's basically it's a schizophrenia drug target, and basically after 2 years of work, sufficient amount, well, like exercising huge numbers of preparation, like the sufficient number of a few hundred like mini data sets. It's can call mini data sets, tiny data sets which like come of a few degrees of rotation obtained in such a scan, and delivered the structures at 3 and 4 that 3.5 to 4 angstrom resolution. This basically the applications of this method are extremely laborious. It's do not know how many of those are we still have to expect. So it shows that it is possible to apply these extreme approaches to some of the targets which are worse, but the effort coming into that is very substantial. So the applications of serial crystallography for time-resolved experiments appear considerably more accessible and more useful, and having a much broader range of targets. So the most like don't want to say the simplest, but the very like efficient way, well known, is sample delivery via the viscose jet extruder. So basically, we can run this the extruder with synchronized with laser pulse delivered on the delivered on a jet to 100 microsecond with the shortest exposure time, which delivers the data until now was half a millisecond for single delay time between the laser pulse and the exposure and the X-ray exposure. We can also exercise now something like half a millisecond. The series of the time series after the X-ray pulse we can collect at 12 is down to 12 kilohertz rate until now. This is a very well running experiment. Probably part of it has been probably discussed by Valentin Gordeliy this morning. This is just one example where we kind of fetching the L state of inverse proton pipe and a 500 microsecond exposure. We are fetching a whole series of a decay of M state with 8 millisecond exposures spanning 10 to 100 millisecond range. We can even quantitatively refine and basically correlate the occupancy of the M state with the spectroscopic response of the protein to that laser point, so provides a very rather detailed and fairly accurate, in combination with the high resolution single crystal data, picture of rhodopsin functioning in this condition. So, well, this is again, probably this is the simplest of all experiment with all time resolved crystallography experiment with the light excitation. There's a very long-standing interest to do similar things on enzymes, and it turns out that on average, from the data compiled by these people some time ago, on average the enzyme turns around, the enzymatic reaction turns around in 70 milliseconds, and there's a huge range and huge amount of targets to millisecond time resolved crystallography, but also that needs to be extended to the much longer time. And basically, with this type of experiments in mind, addressing the time resolved studies of enzyme catalysis, we've built a dedicated station, which is the first one dedicated, like a synchrotron instrument dedicated to millisecond time resolved macromolecular crystallography. So basically, what we are doing is that, like, while the standard time result standard high throughput experiment is being run here for 6 days in a week, on the same time, this area, the second experimental hatch is used for preparation and for setup of a time resolved experiment, and when it's ready, we're mounting the mobile transport pipe and transport the beam into a second hatch and perform the time resolved experiment. In this way, basically, the instrument is used full time and combines in efficient ways the standard high throughput and time resolved application. So the workhorse sample delivery method at T-Rex station is this patterned chip filled with like with samples. So one assumes that the pattern should, well, basically the crystals should fill this regular hole pattern. The crystals often disagree with that, but you see that at higher zooms that the one third of compartments are very well populated, even in such an unfavorable crystal habit case. And then basically what happens next, there's a piezo micropipette which can deliver nanoliter droplets on demand onto every compartment. So basically this is a typically substrate applied to a crystal in any pretty much arbitrary timing pattern. Yes, so basically we can access them by this after applying substrate to a crystal, which we can access this crystal like after a millisecond or after a minute, or both, even in the same experiment. This gives a very basically high flexibility in terms of experimental conditions to be realized. So as we understand that we need to keep our crystals hydrated, therefore basically there's a controlled environment box built around that experiment, which in addition allows not only control and adjust humidity for stabilization, for crystal stabilization, but doing it at a very broad range of temperatures, basically as we need for characterizing enzymatic reactions. So that's a result of it. That's basically the experiment of xylose isomerase done at 4 different temperatures at 5 different time points, rather slow reaction at this time. So the enzyme is very slow at room temperature. At 40 degrees, which is the temperature of the bioreactor, this reaction is an important step. Basically, that's the way how most of the sugar, the consumer sugar, produced around the world, not in Europe, but in other parts of the world. Basically for making sugar out of the maize syrup, and basically, so the result of it, we can see how the binding proceeds, like versus time or versus temperature, and how the reaction proceeds, how basically the reaction proceeds and the substrate released essentially in the same way as kinetic, as kinetic is measured, just with the difference that the response in this is directly the observation of structure, of we are directly looking at the binding and the reacting at the atomic scale. So this is a unique possibility for studies enzymes and applied very actively. Say we're running 1 day per week now such experiments since last 1 to 1.5 years. So it's an interesting question that we also try to address. Is basically is the data quality, and in our serial crystallography experiments, and what limits this essentially. And we're able to carry out experiments on 2 systems at literally identical preparations and very similar conditions in terms of sample delivery at using XFEL serial crystallography and synchrotron serial crystallography, come up with almost identical results in terms of the data quality and resolution, which basically demonstrates that the data quality is defined by the quality and the amount of the sample used in the experiments, rather than by X-ray conditions. And this point also that basically that the radiation damage we do see the significantly more radiation damage in synchrotron experiments, but it's this data quality, the structural damage is beyond interpretation. In fact, but if we start comparing in the same time the quality of the serial crystallography data with the single crystal data, in this case, for an example of myoglobin, we see that basically whereas single crystal data provides us perfect display of perfect sensitivity to the atom time, correct binding geometry, correct binding distances. This is rather far from being as precise as single crystal data in serial crystallography experiment, and there's a lot of work still to go into our serial crystallography methodology to provide the data comparable, or to the data inequality as we need for the level of details we want to extract on basically on protein structure in dynamic studies. So basically, what is that level of accuracy that we can achieve now with a single crystal diffraction method? And I think basically this is directly contribution of the high brilliance and like synchrotron sources. This data the data of this type were only available previously for a small number of rather simple and small small system. What we see now that we can actually systematically do like an experiment on rather complex and elaborate system, which deliver, like us in these two examples, the direct display of a shared photon. Everybody knew before these experiments, which were done at P14, that basically that the single well shared hydrogen, shared proton and single well hydrogen bonds exist, but basically here we provide a direct display of it. So when we can see the basically previously unseen and unsuspected chemical linkage of lysine to cysteine in this enzyme, which is redox sensitive, and basically this is this is a mechanism of regulation of this enzyme, as well as a bit more complicated story, which I skip for the lack of time. We can see the substrate distortion which basically would not be foreseen, which basically would not be considered as possible on the on a substrate enzyme complex, which explains a lot of enzyme catalysis, and some of the very basic catalysis theory needs to be reconsidered. There are actually three components to this improvement, three important instrumental contributions to this improvement in the data quality. First one is a tailored top hat beam. So the problem is that for high resolution, high accuracy, high precision crystallography, the synchrotron which has to be done on the crystals of few hundred micrometer size, the synchrotron source is much too small, especially in the vertical direction, and there was no way for a long time, no way available to form the coherent beam on the sample position. We basically approached this by using lens optics instead of mirror optics, and putting the image of the lens on a sample instead of putting the image of the source. So we can provide the beam of our bit, well, like within a reason, we cannot go too big, but within a reasonable size, we can define the beam as required, completely homogeneous, and like basically we do fulfill the very basic assumptions of single crystal crystallography from a textbook. This is a strong contribution to the data quality. Well, but I also skip that example for the lack of time. The second contribution is a high high atomic number detector sensors, which became available in a very very recent years. So it was known for, well, again, from the basic, very basics of the crystallography, that high energies are advantages for data collection, in particular for high resolution data collection. And basically this, the application of this has been called by availability of efficient detectors for this energy range. Those became available with sufficient quality and sufficiently large dimensions in the last couple of years. And the third contribution is basically the methodology of the data collection that we are using for that one, and integration of that. This basically is based on the of so-called workflows developed by our colleagues in Cambridge from Global Phasing Lab by Dmitrii Zabelskii, which boils down to collecting like not a single scan, but collecting on every crystal as set of carefully selected scans at various sample orientation, which delivers actually the best possible sample of reciprocal space, the best possible sample of those, and minimizes the systematic errors in our data. And basically, to come to the end, I would like to show the kind of data that we like to collect. That we are collecting now. So this is 560 collected on the 560 kilodalton complex at 1 angstrom resolution, and you see this is interesting numbers. That this is a data set with 3 million unique reflections in the data set. So some part of crystallographic methodology, very basic like tools in crystallography need to be revisited in order to be handled such data, but it provides unprecedented accuracy and details of a huge complex. And basically, to finish here, I would like to say that this is, in my view, this is the future of crystallography. Of synchrotron crystallography. We will shift from like producing a large number of structures to producing a very high precision and very high accuracy structures, which will allow chemical which will bring in the information on the enzyme chemistry, which is not accessible to either electron microscopy or to artificial intelligence based modeling method at this point. And basically, so we will keep on using all our beautiful high throughput kit for the years to come, but the focus will definitely shift from quantity to quality in crystallographic data. And I would like to acknowledge a big amount group of people contributed to the beamlines and its application. And thank you for your attention, and I'm happy to take a question if there's any.
Okay, thanks, Gleb. So unfortunately we are a bit out of time, but I guess everybody understands that you are fully equipped there. So very impressive. Thank you so much. Okay, colleagues, we're going to the last but not the least speaker, also from Germany, from near the same place. It's also about the state of the art, but unfortunately unavailable for us infrastructure. So it's Dmitrii Zabelskii from European XFL. Dmitrii, are you here?
Yes, can you hear me?
Okay, yes, we're here. You, so please take your time.
Okay, let me share the screen. Wait a second. A little bit differently. 1 second.
Yeah, it's okay.
Okay. Okay. Hello, everyone. My name is Dmitrii Zabelskii. I'm a postdoctoral researcher at the European XFL in Hamburg. And to start with, I would like to thank the organizers to invite me. It's a great opportunity. And also many thanks for giving me a time slot right after Gleb, because it's a great opportunity to kind of stand on the shoulders of giants, because my talk will be in many senses about comparison of XFLs and synchrotrons, which are doing similar things, but a little bit in a different fashion. And I also would like to thank Dr. Adrian Mancuso. It's our group leader who is now moved to Oxford, who is actually author of many of these slides, and I'll be presenting results of his group mainly, and give an overview about how XFL works and what we're doing in Hamburg in this direction as well. So just to start, there is a short overview of what I'll be talking. I would like to cover multiple topics, and I will not be able to provide detailed overview on many. So if you have questions, please approach me and I can discuss more. But essentially what I will be talking about today is what are exactly free electron lasers, FELs, and why are they different and how are they different from synchrotrons. And I'll be talking about how X-ray FELs are enabling structural biology research, which is a little bit different from synchrotrons and what could be achieved there. And I'll be talking more specifically about European XFL, which is in Hamburg, and what this particular XFL difference from other XFLs. I'll be talking about what are possible differences and difficulties of XFL science from synchrotron science, and also cover advantages and disadvantages of XFLs and synchrotrons. And I really hope that Gleb will agree with me at the end of my talk. So why do we need XFLs, or what are XFLs? As Gleb also explained in the previous discussion, so synchrotrons are pretty much able to produce essentially X-rays at a certain rate and a sort of big brilliance. And XFLs are different in a way that they produce much more spatially coherent, compact, and more brilliant, which is a key part, X-ray pulses. So if you compare synchrotrons with XFLs, you can see that XFLs pretty much capable of producing much more brilliant X-ray pulses, which brings both advantages and disadvantages. And those are femtosecond duration, which is pretty important for some applications. Yeah, they're high energy. And also those are hard X-rays, which enables structural biology for us. So XFL pretty much offers possibility complementary to the synchrotrons, but this will be a recurring topic for this presentation. So just to summarize, ridiculously bright, ridiculously short, and spatially coherent. And all of those properties are key for XFLs. So just to give you a small overview, there are not too many XFLs in the world. So as far as I know, there are about 50 synchrotrons. And I mean, many more than the only 5 of the hard X-ray FELs in the world. And some of those are getting into the reconstructions from time to time, and XFL is one of the newest ones, which is actually started operation in 2017 and only somehow getting started in some sense and getting acclimated to more users. But there are not too many, and this is in some sense unique science what is done with XFLs. And since I'm from European XFL in Hamburg, I'll be mainly focusing on the European XFL. So one important thing is how exactly the X-rays are delivered. So the most established and well-known XFL in the world is LCLS. This one is South California. And they are now undergoing an upgrade. Essentially, they are building one more XFL, which will be LCLS2. And they will be going to 10 hertz to 100 hertz repetition rate, meaning that essentially 100 times in a second they will be delivering a pulse. And European XFL is drastically different because in European XFL we have a completely different time structure of delivered pulses. And pretty much every other XFL follows the LCLS approach, just delivering the X-ray pulses once in, let's say several milliseconds. And just to give you an overview how XFLs are different from synchrotrons. So synchrotrons are essentially big rings, and XFLs are not the rings. Those are giant accelerators. So for instance, European XFL consists of 3 sides. So all the end stations and the instrument are in Schönefeld. So Desi-Barnfeld is accelerator side, and Asdorfer-Born is another patch with a essentially accelerator side when the beam is spread into the different instruments. So about the time structure of particularly European XFL. So this is a pulse source, spatially coherent, high pulse energy, everything I mentioned. And what's most interesting about it is how exactly XFLs are delivered. So you have each 100 milliseconds, so 10 hertz repetition rate, get a pulse of many femtosecond short X-ray pulses, which are delivered at 4.5 megahertz repetition rate, which allows much faster potentially, and also for some application important delivery of X-ray pulses. So there are 27,000 per second, which is absolutely incredible achievement, but it also presents many difficulties, both in terms of the structural biology, material science, and everything. And yeah, there are many key ideas which I will cover in later regarding this megahertz repetition rate. So there are 6 instruments in European XFELs, and I have to say that that's also a key difference of XFELs, that on a typical synchrotron, you have tenths of the end stations, tenths of the instruments, so around 30 on average, but the number might vary. And on XFELs, you have much fewer number of instruments, and so there are only 6 of the instruments, and only 3 of those can be working in parallel. So, which also limits a little bit the possibility of getting the beam on the XFEL in general. But the beam, in a sense, is much more interesting, if you can say it that way. I'll explain in more details what it means later. So now I'll be talking mainly about SPB-SFX instrument, which is the structural biology dedicated instrument. And we are doing many things, so the scope is extremely broad for XFELs. So probably the most important powerhouse is the serial crystallography, which is bringing the most number of the scientific cases and probably the most successful part. Then there is a small angle scattering, which is also possible at XFELs. And probably the most interesting thing is how you can see the things at the femtosecond resolution, which I'll be talking about a bit later. Single particle imaging is another very important scientific case, because usually for scientific, usually have a concept of a signal to noise ratio, and it's only possible to study crystals, which essentially present itself a repetition of the same molecule many times, and then the signal is amplified by the number of proteins in a unit, or whatever is in asymmetric units. But at the XFEL, because of this ridiculously bright structure, you can actually study single particles. And it is done very well. There is established cases for viruses. And it's pretty interesting how you can just take one molecule and study it at such a high resolution. So the maximum resolution is about 20, if I'm correct, angstrom, which is great for viruses. So it's pretty interesting case as well. And the last but not the least, megahertz microscopy, where you can image your sample from many different dimensions, from many different angles at the same time, and you can get many processes at extremely high, both temporally and spatial resolution, which is interesting for fundamental science. I'll be mainly talking about serial crystallography and achievements in that part, since it's mainly related to my work, but I encourage you to ask about other cases if they're interesting to you, probably personally, or yeah. So I'll be happy to talk about different cases as well. So now I'm approaching a different topic. So why do we need XFELs for structural biology, and how exactly are we gonna study our samples? So because of so Gleb previously explained the concept of the radiation damage in biology, and essentially when you have a crystal in a synchrotron and you illuminate it with a beam, then the protein your crystal is slowly dying and is slowly accumulating damage, and for some cases it might be very difficult to get the information from the damaged crystal, and it's like a very important topic in structural biology in general. So XFEL allows you a little life hack, meaning that since your sample is immediately destroyed by XFEL beam of such high brightness, then you only get the diffraction of the radiation damage-free protein, meaning that while destructing your sample, you also collect the information of that, and then you only your detector only gets the diffraction from the untouched, quote-unquote, sample. But it also means that you have to follow a little bit different approach. So you can't just rotate your crystal as in a synchrotron usually does. But you have to deliver a sample differently, and then you have to merge and collect more data from crystals. So the sample is usually delivered by a liquid jet or other means, but not with a goniometer as it is done at the synchrotron. Also, X-FELs are uniquely suitable for small crystals, and that's probably one of the key applications for XFELs in science, because small crystals are rather often not really accessible by the synchrotrons because they can't really tolerate enough beam and they don't diffract to the higher resolution because of how small they are. But XFELs are much more suitable for small crystals than big crystals. So if you have big crystals, you really need to go to the synchrotron rather than to the XFEL. But small crystals are bread and butter of XFELs. They are both necessary and in some cases can only be studied at XFELs. So that's pretty important feature. And the last but not least, this femtosecond timescale, which I was previously talking about. So Gleb showed the amazing videos of how they were able to get the later stages of the photocycle, this inward proton pump. And so when you're looking at the, let's say, microsecond and millisecond timescale, then you would like to go to Gleb and talk to him about how they can measure your crystal there. But if you're looking at the much shorter time scale, let's say femtoseconds, picoseconds, synchrotron just doesn't cut it because of much wider spread beam. So they just can't deliver the femtosecond pulses. But this is possible at XFEL because of its intrinsic properties. So essentially, you can just, for the photoactivated system, but also for the other systems, you can just shoot the optical laser into your sample right before it is hit by the XO beam. And you can get the precisions up to tenths of the femtosecond, which is pretty amazing how I see it. Okay, so now I'll be talking a little bit more about this PBS-FX instrument and what are we doing in Hamburg. So here's how instrument looks like. So you can see the detector, you can see a sample chamber, and you can see one of my colleagues who is doing some work in a sample chamber. And there is a pretty wide range of energies which can be covered. And what's probably most important is how the sample is delivered. So it is delivered by the liquid jet, and there is many different approaches to the liquid jet delivery, and that's one of the most essential parts of any experiments. So if your sample can be delivered by the jet, then you should probably try going to European XFEL, because then you can collect your data very fast and very efficiently. So here is just a reference for the instrument itself. And it is a minor miracle that we can do experiments at megahertz repetition rate, because everything should be not only XFEL sturdy. So XFEL is pretty much very dangerous because how intensely bright it is. And because of the megahertz repetition rate, you should be able to collect, store your data. And also your hardware should be capable of handling this megahertz repetition rate. And it's not an easy task. I will not go into many details, but I just want to tell that it's extremely difficult how everything was brought together. And now it is all working in a pretty much symphony. So it is amazing. So now we'll just be showing the typical experiment, how it's done. So once again, you have this burst of X-ray pulses, which are brought at the megahertz repetition rate. You have your sample, which is essentially focused at the X-ray pulse. And then if you're lucky enough, your pulse will hit the crystal, and then you'll get some image of the detector. So here's the schematic of the very first experiment. It's pretty much the same. It's a common one. And because of the time pulse, because of the pulse structure, so you hit, your jet is hit by this high repetition rate, and it is a question whether it is too fast, and then you probably hit it with the only first pulse, and all the later will not be seen. But if you actually take a look at the real data which we're taking during the experiments, you see that it's possible to hit different parts of the jet and still collect the data. Okay, so those are key questions. I pretty much said them. And here is the very funny video, I find it. So this is the liquid jet with a sample, which is hit by the X-rays one of these trains. So the sample delivery is very fast, and that's why I'm saying that it's very difficult to actually put your crystals into the liquid jet, and it's the extremely important part of the preparation of any experiment. It's not easy to put your crystals in such a jet, and also it consumes a water sample. But what's important here is that the jet actually reforms before the next pulse arrives. You can see that you get this nice structure, and everything works, at least on this video, pretty effortlessly. And if you reduce the speed, you still get your jet running. And so everything pretty much works with the sample delivery if done right, and you're actually capable of getting the pulse of the XFELs and collect data in the detector. So jet reform is necessary, but not sufficient condition for zero crystallography. Okay, and so we collect a lot of data. There are many examples. So here you see Bragg peaks, and that's great. Yeah. And as Gleb also mentioned, we are capable of producing high quality density, electron density maps, and it's actually possible with a megahertz repetition rate. And again, this radiation damage-free concept comes into the play. And yeah, so you see many important parts not affected by the radiation damage, and sometimes it's very difficult to study such systems at the synchrotrons, but X-rays, IXFELs allow to study such systems just as it is, as they are. Okay, so here is some examples of the more important science which were done at the European XFEL. So this project was a part of a huge collaboration and to tackle the problem of the COVID pandemics, and they did analyze the data from one of the COVID proteins, so it was protein M, and the data was collected at the XFEL as well. And it was possible to analyze how the structure-related function, how the structure affects the function of this protein, and can we actually find new sites for potentially drug discovery. So it's a pretty common case of the using the structure for the drug discovery, and in this case, it has a pretty important meaning to reduce the losses from the COVID-19, which is global pandemics apparently. And yeah, another pretty important case for XFEL science in general is this eco-friendly exenticide. So there are some bacteria which produce their toxins as proteins in nanocrystals. It's very small crystals essentially. And those crystals are produced not by the man, but by the bacteria. And because they're very small, they're actually very suitable for XFELs, but not for synchrotrons. So those are tiny crystals, they're native, and they have many applications because they're toxins, apparently. And people are interested in those because it's pretty much the only way how you can study those. And yeah, XFEL is a great option to study. Okay, so once again, one of those components, and there was a description of the particular experiment which was used for those crystals. It was one of the remote beam times at the XFEL in Hamburg, and there are some parameters how they studied. So they used nanofocus, which is probably the best options for the very small crystals. And yeah, so here's the preprint, how it was done. Here's the example of the sample delivery, which was used for particularly that experiment. So as I say, the sample delivery is one of the critical parts. It's very difficult. And in this particular case, the sample was covered by the additional layer of liquid, which is usually ethanol, and then you have a focusing gas. So it provides this additional focusing and jet stability, reinforce the jet stability, essentially. And then, as you can see, you're capable of getting this very precise jet. And this is the static photo, but jet was pretty much stable. And this is also because of the work of the sample injection team at European XFEL. So here is once again the double flow focused liquid injector, which is now widely used at XFELs as well. Okay, some more. And yeah, so what you know from some other talks in this conference, AlphaFold is an answer, but not an answer for everything, and it will not get us all fired, hopefully, at least not too soon. And so some of the structures of this bacterial insecticides were only accessible with the AlphaFold, but not with the means of structural biology. And here is an example when the XFELs and these tiny nanocrystals were able to actually match in some sense the AlphaFold and get the structure, very new structure of these bacterial toxins. So yeah, structures in 1 and 5 were only accessible with the AlphaFold, and there was one particular beam time when you can see that people were able to collect multiple structures in one run, and that was the example of the very well prepared experiment and very successful as a result. And I really hope that they will be successful in developing drugs for those toxins as well. Yeah, 5 structures from a single remote beam time, it's actually excellent result, so to say. Yeah, here is the print screen. Okay, and one more topic that I want to discuss is the palm probe experiment. So some proteins are intrinsically photoactive, some proteins can be used with the cage compounds. So there is a lot of interest in studying the photo processes and correlation of the XFELs with the optical laser. So here is an example how it can be done. So essentially have 3 axes, and you can add to the sample delivery and the XFEL beam the optical laser, which can be correlated with the beam delivery essentially, and then you can get the femtosecond scale resolution of your time resolved structures. And I'm not going to go into many details explaining how it works. I just want to show you an example. So yeah, the XFEL, you can pretty much get it done in the picoseconds and hundreds of femtoseconds. And European XFEL is particularly different because of its pulse structure. It also provides you with this additional microsecond resolution. So there is a 4.5 microseconds between the pulse trains, and you get them just as it is because of the structure of the beam itself. So it allows this intrinsically possible time resolved experiments on a microsecond scale, which is actually a great complementary tool to the synchrotron data. Yeah, and probably one of the last topics which I will cover today, one of the last examples, is the mix and inject type of the experiment. So you can not only do the time resolved optical laser delivery, but you can also mix your crystals with some small compounds and see in a real time how the substrate can be bound to the protein. And here is one of the examples. So there is an enzyme, beta-lactamase enzyme, which is actually pretty important in terms of the antibiotic resistance. It's one of the, so the small compound is essentially an antibiotic which is bound to the one of the bacterial proteins in order to prevent bacteria from multiplying. And it's a studied process, but people didn't know the dynamics, and it's an excellent target to get this interesting data. So once again, they're pretty much the same experiment, and I have a very nice video. So here's the example how the inhibitor appears in the binding site of the protein. And you can see here it's a millisecond time scale. So in some sense, it's accessible for the synchrotrons, but it's a little bit easier to do with the XFEL because of the more developed, I should put it in quotes, sample delivery infrastructure. But essentially, it's also another example of how we can do time resolved business using X-rays in general. Okay, so I'm approaching the end of my presentation, and I just want you to remember a few key things, these take-home messages. So first of all, it's a wide range of science which is possible at XFELs. So I pretty much covered only one small topic out of the big range of possible topics which are possible with the photons and XFELs in particular. And also there is a huge chunk of the material science which is extremely interesting, and it's also accessible at with XFEL sources. So just to remind you, even though I repeated it 5 times already, XFEL pulses are ridiculously short and bright, and it's enabled the collection of radiation damage-free diffraction from small crystals. And small crystals are the key target for XFELs, and often, not necessarily always, they're not suitable for the synchrotron. XFEL pulse structure allows pulse probe experiments with a femtosecond timescale, which is often biologically relevant. There are biological processes such as this retinal isomerization, which was discussed previously in rhodopsins, and it can be only probed at XFELs, and it's pretty biologically relevant and interesting. And particularly European XFEL in Hamburg has a unique pulse train structure, which allows much faster collection of data and also gives you this additional time resolution for the time result experiment, this microsecond resolution, which is particularly interesting in some cases. And here I want to a little bit add up to the summary that XFEL science is largely complementary to science in the synchrotron and is usually used for specific scientific cases. So as I discussed previously, there are not too many XFELs in the world, and there are not too many instruments on each XFEL, meaning that the XFEL time is usually very, very expensive. And also there should be a very specific scientific case to study crystals or other targets at XFELs, meaning that XFEL data usually requires you first to go to synchrotron and actually see where they can do things at the synchrotron. And for the many cases, crystals are much more suitable for the synchrotron radiation science than rather than the XFEL science. And because it's so difficult to reach XFELs in some sense, and you should have a specific scientific case for that, then people rather often just stick to the synchrotrons. But there are also the scientific cases, which I just described, which could be extremely suitable for XFELs, and I really hope that there will be many more experiments done in the future. And just to mention that XFEL experiments usually consume a lot of time in preparation, a lot of sample, and they're not easy by any means. But if the results are achieved, those results are of excellent quality and very publishable. Okay, so I just want to thank the SPBSFX team. There are many people, and particularly our ex-group leader, Adrian Okuza, who is now a scientific director at Diamond. So there is a slide for funding. So many people give money to XFEL. It used to be, So thank you for your attention. I'm happy if there are any questions.
Okay, let's thank Dmitrii. We have time for a couple of questions, colleagues. В чате-то?
How close is your, okay, can I read, right?
Okay.
How close is your current technology to measuring diffraction from single protein molecules? The dream of about 20 years ago. What's the current bottleneck? So there are many bottlenecks, and okay, so let me say first that there is a paper coming from also our group when it was done by our simulation guy, who's done an excellent work on analyzing SPI data, and there are many limitations. So it's essentially similar to the discussion in cryo-EM. What's the maximum resolution of the cryo-EM? And the idea is the maximum resolution estimated by now is about 10 angstrom. And for many proteins, you don't really need 10 angstrom resolution. So it would be very interesting in terms of the viruses, let's say. But for the single protein, which are also often intrinsically, so they're intrinsically flexible, it's not really possible to get the super high resolution. So far as I know, the resolution typically is about 30 angstrom, so 3 nanometers, and it could be improved to 20, but not too much more. So 10 is kind of estimated limit, and in many cases it's sufficient. But I don't think it's possible, at least with the current generation of the XFELs, to go any further. I hope it answers the question.
Okay, Dmitrii, I have one.
Yeah, thank you. Thank you very much.
Thank you for the question.
Yeah, I have one more question, also concerning this single particle analysis. Just comment, am I right or not? As I understand, if you in this case, you will get the continuous diffraction, and so this means that you don't need to solve the phase problem. You can get phases directly from the images. Is it right or not?
I think you still need to get the angular distributions, but essentially it simplifies a little bit your problem. So I would encourage you to take a look at there is plenty of SPI published papers on mainly viruses, and because viruses have this symmetry usually, which simplifies the case, the scientific case, then the data also has this, additional symmetry, so you don't need to collect too much data, and then you follow the same angular reconstruction procedure as you do in both cryo-EM and, for instance, so XFL data in general. Yeah, but I think you still need to solve the phase problem, but it will be a little bit more simple than in case of the crystals. But there are plenty of examples of the real data which is treated and they describe the procedure. Yeah, so I encourage you to take a look at it a bit. Okay.
Any questions?
Sorry, it's probably not exactly what you wanted to hear, but yeah.
Okay, so no more questions. I think we need to process all the data for this day. So thanks a lot, Dmitrii. And
thank you for inviting me.
Yeah, bye.
Bye.
I can see in your eyes that you are ready for a dozen of more lectures, yeah? But unfortunately not. So we have to close. I would like to thank once again all the speakers for today. It was quite interesting, quite useful for sure, and hopefully not very boring, yeah?